Thomas Durkin is an associate professor at the Montreal Neurological Institute, also known as the Neuro, and McGill University. He's also director of the Neuro's Early Drug Discovery Unit, focused on the use of human-induced pluripotent stem cells for fundamental and translational discovery projects through partnerships with academia and industry. Founded over a decade ago, the group has established a cohort of over 220 iPSCs that have been advanced into different projects within the group and used to generate a wide range of neuronal and glial subtypes, in addition to more advanced 3D brain organoid models. Okay, let's make sure your slides are up and running.
You see those slides?
Yeah, it looks great. Floor is yours.
Okay, manage this. Okay, thanks everybody. Nice to meet you all, and thanks for coming to the webinar. It's great to be here today. I'm very happy to be talking about one of the projects we're doing in the group on how we're using iPSC-derived brain organoids, in particular midbrain organoids, to model Parkinson's disease in a dish. So, as Irish mentioned, yeah, I'm actually from the Neuro, so McGill University, and I'm also heading up what we call the Neuro's Early Drug Discovery Unit. Okay. So what we do is kind of like a full circle approach. Like we work with patients. Patients come through the neuro, and then we actually use their cells, so the blood, to then reprogram into an iPSC or a stem cell. We can use it from there, or we can actually CRISPR edit it to then correct the mutation, to introduce a knockout, even reporters if needed. And then we have a whole quality control process that then leads us to then use these to make disease relevant cells for assays that we can then onto our high content and automation platform. And that's kind of where the emphasis will be today, on how we use these cells, kind of neurons, glial cells, organoids, to then build 2D and 3D models of brain disease in addition. The goal is really hopefully to bring kind of, to test drugs, therapeutics, RNA, to see what works, maybe what doesn't work, and eventually then to bring it back into the clinic. So we started about a decade ago. We kind of started to work first on dopaminergic neurons, and over the last kind of decade or so, have really kind of morphed into other models. For projects with ALS, we do make skeletal muscle. We can do dopaminergic, cortical, spinal. We do different types of glial cells. But really, the focus for today will be more on the 3D model we do, and in particular, the midbrain organoid stuff over there. But if you have a C model, you want to know more about what we do on it, please feel free to reach out at any time. So the reason why we want to kind of make these organoid models is we have a lot of patients that come through the door within the neuro who with different types of Parkinson's disease, and they always have the caveat, or they always have the saying when it comes to patients with PD, when you meet one person with PD, you've met one person with PD. It's a very heterogeneous disease, but it's often always grouped under this umbrella. So to really kind of get a sense of it, we're trying to, how do we get as many lines as possible to make as many brain cells as possible, more kind of physiologically brain models in adults, to then understand what might be causing the disease. And we really focus on two main keys here for this talk: what they're made of, so the single cell features, how you can break them apart and start to kind of almost stitch them back together like a jigsaw, and also what do they look like, their morphology, kind of through immunofluorescent imaging, morphological imaging, and then more 3D reconstruction imaging, which we've been starting to do more recently. So when we think about Parkinson's, we always kind of think about one or two of the features, the postural disturbance, rigidity, tremor, but there's a whole bunch of stuff that's below the surface. It's like you kind of see the tip of it like the iceberg, and there's a whole bunch of stuff that goes below it. Often, actually, one of the earliest signs of Parkinson's is sleep difficulties, anxiety, depression, but it often kind of gets missed, even loss of smell. And so, in a way, it's like when we get patients with Parkinson's, we're actually seeing them almost 10 years, 15 years into the disease. So, what is really going on at the earliest stage? So, that's kind of really where you start to need to build better models to really understand why the disease is happening. There are two main kind of features of Parkinson's, really. A loss of these dopaminergic, or these dopamine producing neurons in the substantia region within the midbrain. As the disease progresses, you start to see a progressive degeneration or loss of these specific neurons within this region. And so gradually you actually start to see within a sagittal section of a patient where you would actually have a lot of this black pigmentation, where the neuromelanin is kind of being produced by the dopaminergic neuron. It's actually gradually lost then within the Parkinson's disease itself. And so this just kind of shows you the circuit of how it all connects back within the regions of the brain. But the question is really then the neurons in the substantia nigra are being lost, but what is this region actually in particular? Could we model it in a dish? And so this is actually where we kind of wanted to first, or others have been really kind of pioneering this work through single cell sequencing with postmortem tissues on patients. There's kind of a misconception that with the substantia nigra, you actually have a lot of dopaminergic neurons, but in reality, you actually have only a very small subset. It's actually composed of a rich kind of collection of glial cells, astrocytes, microglia that all come together to really support these dopaminergic neurons. And you also have the GABAergic neurons that are there to kind of help control the activity. You can then actually kind of pull it out. You can see some of the markers like tyrosine hydroxylase, SLC, which really help to demarcate these neurons. But it kind of just shows you how complex an actual this region would is here. So this is almost like what we want to get to. So we have to then kind of go back to the beginning. So what we want to do is when we first started is to study Parkinson's, it's we want to make dopaminergic neurons. And so this is the team that helped us get this, help kind of establish a lot of this over the years, both in terms of establishing protocols, characterizing, and even checking between different lines from different patients. We follow kind of the standard double SMAD inhibition protocols, where you can actually then go from an iPSC, you would then grow with the different kind of chemical factors, growth factors, to kind of a midbrain dopamine precursor that would be FOXA2 positive. We would then kind of expand them gradually a bit over time until eventually then we could bank them and then start to mature them into early stage dopaminergic neurons, so 1 to 2 weeks, and then you can even mature them even longer. And once they're being grown longer in a dish, like even up to 4, 6, 8 weeks from the progenitor phase, you start to look for markers. And in particular, one of the markers we just show here is tyrosine hydroxylase. So with maturity, you then start to see a lot more of them. If you want to then show 6 weeks, this is kind of single cell sequencing just to show you the complexity of it. You do actually have populations where there really are mature dopaminergic neurons. Quite a lot of it still is really more focused on the immature dopaminergic neurons, but you do have some glia, which is not microglia, it's like radioglia, and you do have some progenitors and GABAergic. But kind of just shows you what we're starting with when we're trying to get to study these neurons for different assays. So one of the particular interests actually when it comes to Parkinson's disease is I've talked about one characteristic, which is the loss of dopaminergic neurons. The other is actually the buildup of these inclusions. They're called Lewy bodies. They're actually formed from synuclein. So synuclein was one of the first genes associated with Parkinson's over 20 years ago. And synuclein actually has a tendency to misfold within Parkinson's. So it misfolds. It actually then starts to seed itself. So it can actually take the monomer that's in the wall type form. It can seed it in the prion-like ability and start to form these oligomers, protofibrils, and eventually these amyloid fibrils. And that kind of forms the basis of a Lewy body. And the Lewy body has quite a lot of protein in it, but actually is bound to a lot of membrane. And so this is really what people are trying to understand is how do these form, where do they go, and how do they move from cell to cell? Because as you see the disease progress, you actually start to see the synuclein move throughout the brain and really start to take up a lot more of the cells. What's really unclear is how they actually get in. There was kind of studies over the years that have shown maybe it gets in through endocytosis, exocytosis, how it leaves. And so for us, we actually, many years ago, we actually started working with the McPherson group. So we actually built the protocols in our group to actually make our own synuclein PFF, where you just take synuclein, you aggregate it for about 5 to 7 days, and you get these kind of nice in-house fibrils right here, where they're actually unsonicated, quite long in size, and then you just sonicate it. So once you sonicate it, you're about 100 nanometers in size or less. And then you can then use those for cell assays, mice injection experiments, and even other kind of studies as well. So this actually led to kind of a novel insight, actually working with Armin Bayati, who's now at Harvard, and actually Peter McPherson's group that was published a couple of years ago in Cell Reports that these PFFs are actually really rapidly taken up. One of the things we would add is we'd add it to the media with regular cells, neurons, and kind of wanted to see is how fast would they get taken up by these cells. And surprisingly, it was actually very quickly, almost within 1 to 2 minutes, when you really did this kind of rapid live cell imaging. Armin really then focused in on immunogold EM to could he visualize the PFS, and he could really start to see once you add the PFS into the media, we actually cluster right at the membrane. These invaginations would form, and then it would get rapidly taken up and go straight to the lysosome almost within the 5 minutes. So over the years, we've actually been finding different tool compounds that if you disrupt the lysosome, you can really kind of disrupt this whole process of PFF or synuclein internalization. But once it's in there, you can actually do kind of other stuff. You can actually start to see is does it seed new synuclein aggregates? And so that actually is what it does. So if you actually give the PFFs to neurons in about 2 weeks, and then you just kind of leave it over time, you can actually start to see within about 2 to 7 to 14 days, you actually do get these aggregates to form. So these aggregates are phosphorylated. So it's phosphorylated on phosphoserine 129. And this is a mark of new synuclein aggregates that do form. This is again in collaboration with Armin and Peter. The interesting thing is you get seeding by itself, but if you actually give an immune response, you really kind of drive and accelerate it. So when they added interferon gamma, it would actually stimulate this massive increase in seeding. So in our groups, we've both done both without interferon or with it, and you can start to actually see this kind of drive in synuclein that forms. And the interesting thing, or the cool finding we did find was this, is you can actually really observe the membranes around it. It was actually pretty cool to see all the lysosomes and mitochondria all around it. So for one of the first times, or what we've seen at least in our studies, you could really start to see kind of these Lewy body formations that would form, and then you could quantify it and see that they would grow over time. So the longer that you're left, the bigger in size they get. And that's always been kind of the case, even in terms of the number of seeds that form or the size they would form as well. So in a way, I wanted to set the scene. So in a way, over the last kind of decade, we've done a lot of work with 2D neurons, and then kind of gradually getting into to nucleon studies. But gradually, we wanted to really start to kind of connect this into what we wanted to do in organoid models. So we wanted to switch from 2D to 3D over 5, 6 years ago with a talented postdoc Ngoy bin Mohammed. And the reason for this is we wanted to kind of work on a model that wasn't just stuck on a dish. We wanted to work with something that was more free floating, interconnected. So something with a lot more of the different cell types. Often with dopaminergic neurons, they tend to be more pure, they tend to be more neuronal. We wanted to really kind of capture the complexity of that piece of brain tissue where we're losing the neurons from, and to see could we make this scalable, not just to make one or two organoids at a time, could we make them by the hundreds and have a system that could be easy to maintain with a diversity in cell types, and that it's also customizable. So for us, we really did midbrain, but we've worked with other groups that have done hypothalamic more recently, cortical, and so on and so forth. So once you kind of can balance the developmental cues with the right factors and so on, you can really start to get the tissue type you want. But for us, it was really more a focus on midbrain organoids. But I do have to kind of always capture it from the famous George Box is essentially all models are wrong, some are useful. So when we're talking about the organoids and what we've been doing over the last few years, there's a lot of great stuff that's come from it, but they are still have some limitations. I'm always happy to talk about it at the end. So what we wanted to get to is we can make a midbrain organoid, but we want to get as close to a substantial algorithm. But how actually do they compare when you start to think about it? So to give you an idea, this is how we actually first started making our brain organoids. An orbital shaker, 6 well plates in an incubator. We would just grow them. You grow maybe 30 to 40 per plate. And then from there, you would actually just let it shake, and then you kind of take them out when you're ready to do your experiments. The way it works is we work from human iPSCs, so from a confluent plate of iPSC colonies, you dissociate them, you put them into a low attachment U-bottom plate to kind of form these EBs or an embryoid body. They're grown for about 7 days with kind of neuronal differentiation media, and from there, then you actually then start the midbrain patterning. So we would start patterning and embed them in matrigel. We would put them in the 6 well plates, and then we would start to shake them. So at about day one, they would actually be maybe 400 or 500 microns in size, but gradually over time, at day 50, for instance, they're actually quite big, anywhere from about 3 to 4 millimeters. And these were some of the earliest batches we did that we published many years ago, actually, with the Magna. So you can kind of really see these clear translucent midbrain organoids, and that actually have a lot of the complexity neural rosettes that you would kind of hope to get these organoid models. So when we started to do them, it was again, we wanted to come back to it. What are they made of? What's the jigsaw of all these cells, and what's in them, and then what do they actually look like? So to do this, we had this good approach to do it, but we needed to do it much wider scale. Partly, we needed to be able to easier to maintain them, and we just needed lots of them by the hundreds. And it actually led us to a startup in Nuvio, actually based out of Montreal. So this is in Ngoy Mohammed and Megnam Holland, who really helped drive a lot of this to kind of create this microfabricated disc technology with them, where we could really start to make these organoids probably anywhere from 100 all the way up to 600, 700 per line if we needed to. It's kind of like a Goldilocks approach in a way. You have a large bowl, small bowl, medium bowl, and it's kind of like which size or which kind of power works the best for you. So we often, when we have a line that's well established, we'll work with the 948 ones. And the way it works is you kind of have these kind of center parts. You just have your suspension of stem cells. You would kind of calculate the dilution, you add them in, and then the cells actually start to settle within each of these kind of little wells. It's like a hive. And then you let it sit for about 6, 7 days. EVs form. And then you can kind of bend it over into a plate with media. And then from there, you would actually then put it into the bioreactor here, which I'll show to you. So this would be what you would see within a 2.5 month culture of organoids. They've been growing. You just grow them in a magnetic stir plate, stir them for about 34 to 36 RPM, and then we just do the media change once a week. But once we have them, you can then actually take the organoids out anytime. We've grown them up to 18 months. At the moment, I think we have some cultures that are 9 to 12 months, and it's often we plan them out almost like a mouse experiment where you just say, I'm making a large batch, and some are going to go for imaging, flow, biochemical analysis, so on and so forth. And you can have different lines all grown at the same time. So by doing it, it really let us kind of get this done to scale, and then it kind of let us and kind of provided the means to start asking some questions. So, for instance, one was, are they actually dopaminergic? Do you know the neurons within these organoids, are they actually functional? Do they form neuromelanins? So it actually led us to form a collaboration with a group, Juan Aguirre's group, and actually Maria Lacali and Aurelio. They kind of helped form together with Ludwig to see, could we actually image these neuromelanin within kind of the organoids using this kind of mesoscopy. So we knew with organoids there was two things. They go brown with age. We would just see them kind of go brown within 5 to 6 months. And when we would section through them and stain for their fontaine staining, we would actually see these brown pigments. But the other thing is, if you added L-DOPA or dopamine around 35, 40 days, they again go brown. So it appears that it's kind of a dopamine byproduct that was forming. And so we would actually then start to actually take different organoids and actually start to visualize them. So you could see here from A to E, you kind of see different examples of organoids, and you can actually start to see kind of through the different imaging, you can really start to see these neuromelanin granules that would form or be present within the organoid itself. So it's confirming that yes, dopaminergic neurons are present in these midbrain organoids, and they do appear to be functional because they're actually able to form neuromelanin. And that was a key feature why we wanted to get into 3D, which I didn't talk about earlier, is because you can make neuromelanin, you're kind of actually almost a bit more mature than what you're getting within a dish, because we never really have seen neuromelanin form within 2D cultures unless maybe you could actually induce that to happen. So once we had that, the other aspect we wanted to see is what do the neurons and astrocytes actually look like within the organoid itself. So over the last year or so, through work from Maria and Martin, which is they've really helped pioneer at working with just AAV models, we just wanted to kind of get a sense of, could we build workflows to really characterize the morphology, the size, the shape, well, what is the complexity of these organoids just themselves? So actually, when we have an organoid, we get these AAVs, one expressing synapsin for neurons, or the other GFAP for astrocytes. Yellow is astrocyte, green, oh, sorry, yellow is neurons, green is astrocytes. And then to kind of actually combine them, and then once you have them, you can image them both in real time through high content imaging on our in-house software Phoenix, or you can actually just fix them and then start to see them at a fixed time point. And once you have that, you can actually start to build 3D models, you can render the cells, and you can actually start to see the area covered, the connections. I want to give you an example of what that looks like. So this is really done in partnership with the Cervo. So they actually help provide us with the AAVs and the kind of reporters that we can just add them on. They get taken up by the organoids, and this isn't just the neurons and astrocytes here, but once you have them, you can really build these cell masks. You can kind of get skeletons. You can start to see how did the cells interact with each other. So this kind of images through about at least probably about 40% of the organoid kind of reconstructed. When we do the bioreactor organoids, they do tend to be a bit smaller. They're about 1.5 to 2 millimeters in size, about half of what we used to generate, partly because it actually, with the organoid kind of in bioreactors, you tend to be a lot happier, less apoptosis. But now when you combine it with the imaging, you can really start to capture a lot of the complexities of what these organoids would look like. You can actually get a lot of data from it as well. You can actually look at individual neurons. You can start to capture the complexity. So instead of just looking at a neuron flat, you can actually start to see the neurons and how they are within this 3D environment. How much do they stretch out? How much do they branch? How many branches actually even exist on it? And then you can start to get a lot of different measurements from it. So that's actually kind of some of the stuff we've now implemented. We're actually in the process of finalizing a kind of an analysis paper that then we can start to then segue then into patient organoid analysis then itself. This just shows you some of the actual complex neurons you can get. So Earlier was more of a flat neuron. This is actually a really widespread neuron. You can really start to see how complex these neurons can actually get when they're actually grown within these organoids. You never see this type of stuff within a 2D flat culture. It really kind of shows the power of what you could get with a brain organoid itself. It's pretty cool. The other things you can actually get is you can actually start to extract different neurons and actually start to see how do they all compare relative against each other. Some are a lot more complex, and some are almost just like one dot, two dot, very kind of small and kind of compact. It kind of just shows you the full depth and breadth of morphologies you can really get. This is just from one organoid. You can do this for multiple organoids. You can do this for astrocytes and other types of glial cells. All you really need is the exact reporter marker, and then you can start to kind of extract a lot of this type of data. So in addition to the imaging, the imaging is great. It kind of lets us extract and obtain details around the morphology of the organoids. But the other thing we really need to do is what's the overall composition? What are they made of and how much do they change? And can we do this in a way that's more cost effective? We have done single cell transcriptomics. I'm sure many of you have done that as well, but it comes with a cost. If you're trying to do this at scale, the dollar signs do add up, and it's not always feasible to do this all. So we do have a flow cytometry group within our unit. So it's actually Julien Sirois manages it. And working with Berlin and Thomas, they wanted to see is could they combine the value of organoids with flow cytometry and machine learning to kind of build a more cost-effective process, which was actually published last year called Cell Typer, where all we did was we took midbrain organoids, we would dissociate the cells, and we'd actually use full antibody panels. These were customized so that instead of just saying, okay, this cell is CD14 positive, negative, we'd actually look at the range of expression on the cell and actually start to combine all the different markers and the scale of scale of expression to then start to correlate this into almost like cell clusters and what actually then would be more expressed. Instead of just saying yes or no, it would be more on a gradient level. What this actually led to was actually this. So we actually have three different control lines here. WHAG 3450. Three control lines, three different people, and they were made into organoids for about 4 or 5 months here. And then we did our profile. We could actually then start to build a cluster on each of them just to show how are they actually comparing against each other. They are actually pretty comparable. We could actually visualize most of the same cell types that will be present, from astrocytes to radioglia. You even start to see these kind of amounts of neurons. They do have variability. You're not going to see exactly the same number each time, but it does start to show you how comparable they are against each other from line to line to line. But one of the other interesting things is how similar is it actually when you compare the same line across multiple batches? And so when we actually took our AIW batch and just grew it 4 separate times, so it was 4 different bioreactors, but it was grown at 4 different times where we would seed them, you could start to see how comparable it actually was. Like, there was variability, a little bit in the numbers, but it actually did tend to be pretty comparable, maybe 60 to 70% when you do it across all the different values. But it was kind of giving us heart that like it's not that we're growing one batch and a second batch a month later and everything is completely different. It is kind of comparable based on the methods being used. The biggest actually change we did find that was actually with age. If you age these organoids, you actually do start to see kind of shifts in the kind of cells present and also how they mature. You start to see kind of going from progenitors to neurons later on in time. So you start to see kind of here a lot more radioglia here, but then you start to go more into MPCs here, maybe a little bit more neurons. And then the neurons already start to kind of appear more in NPCs, another cell more kind of in the later stages from 98 to 155. So that was really the key is the organoids, they do tend to be consistent in their composition of cells, but they do shift over time. So there is a change in their identity with maturation over time, these organoids. One of the big powers of why we wanted to do this was actually to get at the cell type of interest to it. So we knew we had dopaminergic neurons in there. It's not present in huge numbers that you really kind of wanted if you wanted to study at it. So to have the cell type meant we could really start to isolate them at scale and really start to ask them questions. So we could actually pull out our neuronal populations, neurons 1 and 2, and we knew the dopaminergic neurons would be here somewhere. And that actually was what we found. So we would take them out, we would sort them, we would cluster them and annotate them. And I'm really just going to bring your focus on to G here. So when you would actually, when this here on top is actually the single cell from the flow, and this here on the bottom is actually the single cell from the actual single cell transcriptomics. You can actually see there's three groups here, dopaminergic neurons SN, substantia nigra, VM, and VTA. And that kind of is shown here in this cluster, but it was able to show is once you could sort and enrich them, you could actually start to see how much of these neurons are actually substantia nigra, VTA, and VM. I'm actually a pretty good amounts of the substantia nigra, but I don't know, anywhere from 25 to 15%, depending on which of the populations. And you do have the other two as well. So now we actually have the ability to make these models. You have the substantia nigra neurons there, but you do have the other neurons, glial cells, and it is starting to kind of get almost a bit more towards the complexity of what you would get within the substantia nigra. So now we kind of have this model. I guess we want to ask is, could we actually use it to even model disease in a dish? So the first question we did is we kind of went back to the beginning. We knew the first mutation associated with Parkinson's was synuclein. So we just asked, what happens when this mutation is present? Synuclein triplication. When synuclein levels go up, what actually happens within the organoid itself? And so this is work, it was published quite a few years ago now. It was actually led by Ngozi Muhammad as well at the time, and many of the other people here in the photo. And she really just asked, we're making midbrain organoids with the substantia SNCA gene triplication. Can we model features of synucleinopathy with the organoids? First thing was, could we actually make the organoids? So we actually, thanks to collaboration with Thilo Kunit's group, we were able to get his line, so an SNCA triplication line, the isogenic line, and the knockout line. So it was really a pair of, kind of a set of three different lines for doing this work. And then we would just make multiple batches of organoids, probably 5, 6 batches to really start to kind of capture both numbers and also any differences that may arise, and then started to see what was going on. One thing we did notice is when you had the triplication line, it tended to be a little smaller by 100 days, but they still grew. So that was encouraging. Even if we had a triplication line, it still grew and kind of grew at a comparable size, even albeit reduced to its isogenic. It was really though when we started to get into the pathology that we actually started to see more noticeable and striking changes. So we knew from the brains of patients with Parkinson's, the more the disease progresses, the more phosphorylated synuclein pathology that gets observed, and that spreads throughout the brain. And so we kind of did start to see this actually with the organoids. It wasn't really at about 50 days. You really had to take this to 100 days. So these are old cultures, and that's another advantage of these organoids. You can actually grow them for a lot longer than with 2D neurons. And you don't really have to worry even about getting stress, attachment as well. You can really grow them and then you can start to visualize features of the disease. When you look at the blot, so if you have the triplication, you have a lot more of the synuclein in the organoids, and it tends to be you have a lot more phosphorylation as well. You would get these little red puncta throughout, and it was increased at about 3 months, and it was significant when you compare the triplication organoid to the isogenic organoid. This phosphorylated synuclein, which was indicative of synuclein aggregates, was actually present in both neurons and in astrocytes, albeit in different ratios. It was almost like in a 2-to-1 ratio of neuron to astrocyte. So it does appear that the neurons or the astrocytes may be actually facilitating some of this transfer. But we actually did verify that this synuclein was also could be observed a little earlier through PLA to really look at synuclein oligomers, and they could also be observed as well. So from this, we actually there's a lot more work within the paper. I'm not going to go into it, obviously, for time, but you could see that through a midbrain organoid, you could actually model features of the disease. You had this first Parkinson's mutation, the triplication, you had its isogenic control. So everything is the same on the genotype, it's just the gene itself has been corrected. The levels of synuclein are actually way up within this line, so that led to more aggregates. And the two other features that I didn't talk about today is it actually led to a decrease in dopaminergic neurons and an increase in gliosis. So you actually started to get more reactive astrocytes. Didn't have microglia yet in it, but this is kind of one of the features we're hoping to explore over time. Well, this kind of did show that two of the main features, more aggregation, a decrease in dopaminergic neurons, all captured within an organoid model. So you always kind of worry when you do work and replicate, because in a way that's the key. Is like 60% of studies often don't work, and so thankfully we actually we're working with a group, Ted Fawn's group, within the neuro. He had a very talented grad student at the time, Ghislaine Diab, who was actually wanted to kind of take up some of this work because as we were winding it down, she was interested in getting it to go. And so a lot of the work we're going to show in the next 2 or 3 slides, really this is all work she really did. So very talented student. If anyone ever wants to meet her. But she really took the process that was in place, making the organoids, but instead of going to 100 days, she actually grew them even longer to go. What happens beyond 100 days? Because when we were doing the study, we were just kind of that was our natural endpoint just to see the data. But it was always a key is what comes next. And this is really where it got striking. So within the triplication, it was cool to see you actually did get these phosphorylated synuclein aggregates that did form. They weren't obviously present in the knockout, and that's an important kind of feature we have to mention for both 2D studies and in 3D. When you have no synuclein present in a knockout line, you can't get aggregates to form. You always need the kind of a seed where you need this kind of monomer to really act kind of to be seeded then into a new aggregate. But it was striking. We actually did see a striking increase in the amount of phosphorylated synuclein within the cells, and it did actually seem to get more intense at about this is at 5 months now. So we've even noticed it from this kind of glance at our study is the longer the organoids are left from 100 days to 170 days, you get more aggregates. And then she actually went up to 6 months. So even then, you had quite a lot of nice pathology. But the real cool feature that she found, or something that we hadn't seen or had started to see, was at the dopaminergic level, where we had started to see a gradual decrease in dopaminergic neurons by ICC, but not really a change in the blood. She took it one step further and let them go to 5 months. And that's really where you started to see this decrease in TH positive neurons. So at 3 months, you start to see a trend in dopaminergic neurons going. At 5 months, it's very clear. So we have more aggregates to form and more kind of a change in dopaminergic neurons themselves. The other aspect is how active or what's going on with the activity. So what we're seeing is more synuclein pathology, less dopaminergic neurons. So are these actually active? So I've shown you a lot of stuff around the features of these midbrain organoids, but I haven't really touched on the last thing is more, do they fire? Neurons fire, they give activity. So we're actually able to, or Ghislaine was, we have an axion in our lab, Maestro, so we can actually take these organoids, put them onto these kind of copper wells, and then actually start to record from them. And it was really actually as you aged an organoid, that's really when you started to see activity. You'll start to see here 1 month to 3 month to 5 months. So with our early work, at 1 to 3 months, you kind of start to see the odd burst, a couple of spikes. But when you got to 5 months, that's really when these organoids have really reached a maturity that the firing is a lot more. You can stop the firing if you add TTX. The firing is almost abolished, and you can record these robustly from batch to batch as well. So it's always a case of maturity. So it is always kind of the best time to then record it. So the question was always, what would happen then if you actually brought the paradigm to the isogenic versus the triplication? And interestingly was, we actually started to get this increase in network burst with the triplication and same with burst number. So it almost was that you were getting a lot more frequency in firing uh, with the triplication versus the isogenic. So you had a lot more spikes in the burst. So that was one interesting feature. And they did appear to be a lot more active. So I'm not going to go fully into the study. If people want to go see it, it's in bioRxiv now. It actually should be hopefully accepted soon for publication. But some of the key features was there was a change in the actual inhibitory neurons, so the GABAergic neurons, and this actually led to kind of almost an increase in kind of how much the neurons are able to fire within these midbrain organoids. So it's a case of you have more aggregates, you have less dopaminergic neurons, and then this kind of affects kind of the rate of firing within your organoid itself. And that has potential consequences for how we're kind of thinking of Parkinson's often with the patients as the disease progresses, there's changes in the movement, changes in the firing patterns, often because of the neurons in the cells being lost. The other kind of key support, I'm just going to touch on another study, was actually work with Christos Gogas in Greece. So actually, we'd been working with him for a couple of years. He has the same line, so it was kind of Gislaine's work really showed the changes in firing and really showed this. Uh, Christos's work really kind of built a little bit differently. He actually was looking more at the ECM. So following similar protocols, he was able to show again you have an increase in synuclein with these kind of same lines in this kind of organoid models, maybe slightly tweaked approach. And you would actually start to, when you would actually take these organoids and build kind of more of a bioanalysis around both proteomics and also kind of transcriptomics, they started to land on ECM genes. So there's actually a change in the extracellular matrix around the organoids. So the increase in synuclein actually seemed to cause certain changes around it. So he was actually able to capture that using kind of a Wisteria floribunda agglutinin, kind of a marker of uh, proteoglycan-rich ECM, and to show that at about day 50 and day 100, you could really, as the organelles are aging, you can really start to see this kind of change within the ECM itself. We're actually almost starting to see that you would get these more wide kind of hollows within the ECM. So the synuclein itself somehow, but because of the changes in it, the aggregation changes in neurons, you're really starting to drive changes within the ECM from the organ. how much of a consequence this has back to the actual patient. That's being investigated by his group now. So interesting to see where this goes. But kind of just shows you through the power of organoids, you start with one study, you get this feature, and then you've been able to kind of show and kind of reproduce a lot of it then over time. So I kind of just want to finish up here just to kind of summarize the talk, just to kind of leave some time for questions. Was hopefully we've been able to convince you, midbrain organoids, they are a good model. They can really be used to model features of PD in 3D in particular, comparable to what you would see within the substantial algorithm. This can be scaled up into bioreactors. It works for some other models as well, not all so far. So, it's still in progress. And you can start to really bring novel approaches both from the imaging and single cell phenotyping to really start to understand the complexities of it. And gradually the goal is really to start extracting more disease signatures from it. So as you start to, now we have a way to scale it up, then to bring in different patient lines. We have our own cohort of GPA lines with isogenics, even LERC lines, TMM175. So to start to kind of make them, compare them, and actually get into sporadic. We have close to 15 to 17 sporadic ALS lines matched with control lines that really start to understand, what may be different. So we do have some further work that's going to come out over the next 6 to 12 months, kind of looking at some more on the transcriptomic side when it comes to these type of midbrain organoids. So take home points. Hopefully at the end of this, with the emergence of iPSCs, we've convinced you can use these cells really to generate human brain cells in dish, like 2D, 3D, what obviously the focus is more on organoids, but you can do a lot within the 2D setup. So if you ever need to ask questions and get the answers fast, 2D neurons, astrocytes, microglia, they give you the kind of tools to really do a lot of stuff quickly. With the organoid, many different cell types can be generated, and when you combine them with the latest methods, either single cell, flow, imaging, you start to be able to observe phenotypes and really start to characterize them in depth that maybe we weren't able to do 5, 10, even 15 years ago. And now with the advent of omics, big data, AI, there's a lot of predictive modeling that's could be done. There's a lot of potential then that can be brought to bear with these models over the next, 1 year, 3, 5, even longer, to really start to understand what is actually causing Parkinson's. And maybe if we understand it, we can then potentially then treat the disease, or at least treat the different subtypes of the disease itself. So I'm going to stop here. This is really kind of just some of the people that have helped with all this work. And I'm happy to take any questions, and thanks for your time today. I look forward to the rest of the webinar.
Thank you very much, Thomas. That was a lovely presentation. We can get into audience questions. So the first one is, a lot of groups have issues achieving high levels of purity for dopaminergic neurons. The question is, what is the level of purity here? And a follow-up question, how much does purity impact the formation of the organoid?
When it comes to 2D neurons, we're always trying to get to anywhere from 60, 70, 80%, like at least TH positive. And then we look at a few other markers as well. I think there's GERK and 2, 3 others. You're never going to get like 100% pure neuronal population. Also, I don't think you want 100% pure population. We've often found that the more we drive the use agents to really remove as many of the glial cells, the neurons are almost less happy or more miserable than they would be before. And when it comes to the organoids themselves, the purity of the iPSC culture is really critical at the start. But is it more how pure the neurons are in the organoid, is it? Or, sorry, I didn't get the last part of the question. Maybe you can repeat it.
How much does purity impact the formation of the organoid?
Oh, yeah, the purity of your iPSC cultures is critical. There's a lot of, it's almost like the first main step. If we're doing an organoid to make it, like we have steps almost in place right from the get-go, that if we see even like a little bit of differentiation, like non-pluripotent stem cells, or if it's almost even beyond 5%, there's questions of whether we go ahead of it or not. So we always try to really clear it up and grow them for 2, 3 weeks to maintain that the cultures are really as clean as possible before then getting into the cultures. And sometimes even within the first days, you'll see the EBs, if they look off, maybe if they look like they're, have extra buds or stuff, you then have to really make decisions on, do I continue with this or do I just restart it? Because these are long cultures. If you haven't got it clean and pure at the start, then you're gonna, lead to maybe some misleading findings down the road. So that was always the key: is that trap goes in at the beginning, trap will probably come out at the end. So you really want to kind of, make sure it's the highest quality at the start.
Thank you. And our last question before we move on to the next speaker is, about using these organoid models to study the effects of small microplastics, so less than 2 microns, and nanoplastics in the brain. Is that possible?
Yes,
It's a good
it's
use of the
possible.
model.
We do it. Well, we have studies that we're looking at. Well, not my group per se. It's another group, one or two. They're looking at environmental toxins. It's funded with the Fox Foundation, more with 2D cultures, not into 3D models yet. But yeah, microplastics is certainly feasible. I know I've thought about it in the past, and if funding was available to do it, we'd certainly get into it. I do think that's an important topic to ask. So yeah, I think if you have an ability to get the different types of microplastics in robust quantities, then you could ask a lot of questions, look at inflammation, for instance, aggregate. So yeah, there's a lot of questions to be answered. It's an interesting question and something, yeah, if we can explore it, I do hope others can at least as well.
Okay, thank you very much, Thomas. All right, welcome back, everyone. Our next speaker is Joanna Neves. Joanna Neves is a reader lecturer in mucosal immunology, group leader at King's College London, and 2023 Lister Institute of Preventative Medicine Prize winner. Neves completed her PhD in immunology at Queen Mary University of London before moving to the Brigham and Women's Hospital and Harvard Medical School to study immunological responses in colitis. In 2014, Neves joined King's College London, where she held a Marie Sklodowska-Curie Fellowship and a RC UK UKRI Rutherford Fund Fellowship before establishing her research group. Neves and her team developed complex intestinal organoid systems that incorporate immune, stroma, and neuronal cells to study cell interactions in health and disease, which led to the discovery of new roles for innate lymphoid cells in inflammatory bowel disease. Okay, let's make sure the slides are up and running. Perfect. Joanna, the floor is yours.
Hello, my name is Joanna Neves, and I'm a group leader at King's College London in the UK. Today, I'm going to tell you how in my lab we study the conversations between immune cells and epithelial cells in the gut in the context of health and disease. The intestine is my favorite organ, and the functions of the intestine go way beyond just absorption of nutrients. The intestine has several compartments. For example, the majority of our immune cells are located in the intestine. It also has a complex epithelial compartment with stem cells here at the bottom of the crypt, have the capacity to differentiate it into differentiated cells, as for example goblet cells, that produces a mucous layer that forms a barrier between our own cells and the bacteria that live in the intestine. It also has mesenchymal cells or fibroblasts that can, for example, have supportive roles for the stem cells, and has a complex microbiota. And the recent survey shows that there are as many bacterial cells in our body as our own cells. This microbiota is composed not only by bacteria, but also fungi and virus. And the intestine also has a complex neural network, including enteroendocrine neurons, vagal efferent neurons, and sensory neurons. So interactions between all of these compartments are essential for the maintenance of intestinal homeostasis. And when this disruption of the interactions, they can lead to both local and systemic disease. So this disruption of these conversations have been linked to the development of inflammatory bowel disease, and we'll look into that today, but also other diseases like cancer, Parkinson's disease, and diabetes. So to study the interactions between the different cell types in the intestine, there is always a trade-off between experimental control and physiological structure. So for example, we can use cell lines that gives a lot of experimental control, but very poor physiological structure, or we could instead use humans ideally, and nowadays using with single-cell transcriptomics and spatial biology, we can infer a lot of information from human samples, but it's hard to do mechanistic studies on those. So for us, the sweet spot is in the middle, is actually using intestinal organoids that give us both experimental control and some of the physiological structure. And I dare to say that when we are looking at the relevance of our studies to understand human biology, maybe intestinal organoids would give us a bit more close to understanding human biology than other models that have mainly been used in the past. So what are organoids? Organoids can be defined as these three-dimensional cell clusters that are grown from either pluripotent or adult stem cells that spontaneously organize into organ-like or tissue-like structures that are enriched for the cell types typical present in the tissue of origin. So in 2017, organoids were considered the method of the year by Natural Methods, and the research on organoids has been exponentially growing over the years, showing the importance of these methods in several fields of biology, including understanding human biology. There's a multitude of organoid models that people can use at the moment. So the first ones were the intestine, both from tumor-derived organoids and tissue-derived organoids, but we also can make them from induced pluripotent stem cells. But nowadays you can think of your organ of interest, and it's very likely that a protocol to develop organoids has been established for that. In my lab, we mainly use intestinal organoids. So to establish those, we either isolate the crypts from the mouse intestine or from patients' biopsies or resections, or we use induced pluripotent stem cells. Then we culture them in very precise conditions in matrigel, and this allows us to grow in this three-dimensional structure that has the different cell types present in the tissue of origin. So for example, we have the intestinal epithelial cells at the bottom of the crypt, and we also have differentiated cells like goblet cells, and the cells maintain the apical basal polarity. This means that the basal side is facing the outside when we be with having our immune cells, and the apical side is enclosed in the lumen of the organoid. So if you think of organoid with like a tennis ball that is very close and the lumen is on the inside will be where you have your bacteria, and then surrounding the outside would be where you have your immune cells. So these figures from my lab where you can see the organoids develop over 5 days. Here they are mouse organoids, and you can see initially is a structure that's semi-round that closes on day 1 and then develops into these beautiful crypt-like structures, or I normally say like these beautiful flowers. So you can see here the video development of the organoids over 7 days. So to establish intestinal organoids from tissue-derived cells or adult stem cells, we isolate the tissue, then we wash and purify the crypts from the intestine, and then we plate them and seed for expansion. These are really easy to manipulate. So after we expand and establish the organoids, we can freeze them and then thaw them to use at a later stage if needed. Nowadays, several countries have biobanks of organoids, and you can actually even purchase organoids from several companies if you want to use them. Organoids can also be established from human-induced pluripotent stem cells, and we also do that in the lab, and I'll tell you about data today that has been done with these organoids. For that, we start with the pluripotent stem cells. We add activin A to induce endoderm differentiation, and then we add Win3 and FGS4 to induce hindgut differentiation. So we check if the cells, for example, express intestinal markers like CDX2 at this point. Then we are able to deform this budding structure, so we collect this budding and we embed it in matrigel like we do for organoids, and then we grow in the same media that we grow for the other ones that contains respondin, HGF, and noggin, and we have the development of these beautiful, again, three-dimensional organoids that contain both stem cells and differentiated cells. So there are key differences between iPSC-derived intestinal organoids and tissue-derived organoids. One of those is the stage that they mimic. So fetal-derived organoids mimic the fetal stage, while tissue-derived ones mimic the adult stage. Nowadays, there are some molecules that we can add to iPSC-derived organoids to promote their differentiation to a more adult stage. And I believe in the next few years, we'll be able to do this more efficiently, and maybe we'll be able to use iPSC-derived organoids, treat them in a way that they could mimic the adult stage, but we are not there yet. So the other difference is variability. So there is reduced variability in iPSC-derived organoids, and sometimes, they are fully characterized genetically because they came from my biobank, and so from what the cells have, and you can have access to their transcriptomics and DNA information. It's easier to manipulate iPSC-derived organoids, but for example, if you want to do CRISPR-Cas9 to knock out the gene, you can do that at the stem cell stage and then grow organoids from that. You can also use CRISPR-Cas9 in tissue-derived organoids. It's just a bit more difficult. The composition of these organoids is also different. So iPSC-derived organoids have both an epithelial layer and also mesenchymal cells, so fibroblasts surrounding this epithelial layer, while the tissue-derived organoids only have epithelial cells. In terms of environmental confounders, the iPSC-derived organoids are virtually germ-free as they haven't been in contact with microbiota, while the tissue-derived organoids are not. And an important note is that both can recapitulate patient pathology. So tissue-derived organoids, for example, from inflammatory bowel disease patients, have some of the characteristics that we can see in the intestine of these patients. And there is a publication that shows that where they made isolate tissue biopsies from ulcerative colitis patients, isolate fibroblasts from that biopsies, induce pluripotency of those fibroblasts, and then made iPSC-derived organoids from that, and they can see that they still had some of the characteristics they have seen in the intestine of these ulcerative colitis patients. So it's quite notable how they can still recapitulate patient pathology. So I start by telling you that the intestine is a very complex organ that has many different compartments, and now I tell you about these organoids that are a beautiful layer of epithelial cells, in some cases also fibroblasts. But what's missing from the initial picture I've shown you? Pretty much everything else. So that's what we have been working in my lab, is adding complexity of these organoids so we can use them to answer the questions that we have, how different cell types interact with the intestine. So we can add immune cells that surround the outside of the organoid to allow us to study, for example, immune epithelial cell interactions, and we will see several of those examples today. We can also microinject bacteria, metabolites of food allergen to the lumen of organoids and to see how they impact these interactions. We can add neurons, for example, enteric neurons to the organoids and see how this immune epithelial nerve interaction has an impact on function of these cells. And we also can play with the extracellular matrix where these organoids develop to see how this matrix and their mechanic properties impact immune and epithelial cell function. And the other way also we can see how these immune and epithelial cells and fibroblasts can impact the surrounding matrix. As we are going to look a lot about these immune organoid co-cultures today, I'm just going to give you an overview of the different approaches that can be used in those. So initial studies were done just by adding cytokines to the media where the organoids were growing. Then follow-up studies were adding immune cells to the media and then organoids still growing in this matrigel. Some of these immune cells could penetrate the matrigel, so it's a bit unclear if all of the cells could have cell contact with organoids or not. For us, our favorite method is having everyone together and embed both immune cells and organoids in matrigel. So in this case, we know there can be cell-cell contact interactions between immune and epithelial cells, and that might drive some of the pathways that we are studying. To clarify if these pathways are actually driven by cell-cell contact interaction, we can then actually do these cultures in transwells, where we have organoids at the bottom and immune cells at the top to see what the pathway we were studying was cell contact dependent. So today I'll tell you briefly about 3 stories where we study innate lymphoid cells interactions in health and disease. In the first one, we studied type 1 ILCs or ILC1s in health and disease intestine. This was driven by Geraldine Jouet, a PhD in my and Eileen Gentleman's lab at the time. Now she's a research fellow at Cambridge University. The second story, we look at type 3 ILCs or ILC3s and their epithelial cell interactions. This was driven by Emily Reed, that at the time was a PhD student in my lab and now is a postdoc at Queen Mary University in London, and also by Enis Penacerra, that was also a postdoc in my lab at the time and now is a lecturer at the Basque University. And finally, the last story is we are looking at ILC development in mucosal tissue, so innate lymphoid cells development in mucosal tissue, a story that actually was started by myself and then followed up by Geraldine Jouet and several other members of the lab. So we mainly focus on the study of inflammatory bowel disease. IBD is a multifactorial disease whose development depends on the genetic of the host, the microbiota, and the environment. IBD can be divided into Crohn's disease that can affect any part of the gastrointestinal tract, mainly the last part of the small intestine, the ileum, and the colon, and can also be divided into ulcerative colitis that only affects the colon. IBD patients present microbial dysbiosis that is characterized by loss of diversity on the strains that have and increase of pathogenic ones. They also have a leaky epithelium, or also known as a leaky gut, means they have problems in the barrier function, and this allows the bacterial metabolites and bacteria acid to penetrate to the epithelium and can drive inflammation. So they have this accumulation of inflammatory cells in the intestine. And the immune cells that we are interested in studying in my lab are called innate lymphoid cells. These are tissue-associated lymphocytes. They are antigen non-specific. They are early factors with rapid and potent cytokine responses. As for other immune cells, they can be divided in different subsets, different flavors, depending on their expression of transcription factors or cytokines. So we have, for example, type 1 ILCs or ILC1s, and NK cells, natural killer cells that can express TBET, or in the other case, IOMIS, and produce interferon gamma. Have type 2 ILCs that express the transcription factor GATA3, and type 2 cytokines like IL-13, 5, and 4. And then we have ILC3s, type 3 ILC3s, and lymphoid tissue inducers that express gamma T transcription factor and produce IL-17 and IL-22. Today we're going to mainly focus on ILC1s and ILC3s. So why do we study innate lymphoid cells in inflammatory bowel disease? These cells, in healthy intestine, the majority of ILCs in the intestine are ILC3s, but in the inflamed tissue of IBD patients, there's a dysregulation of ILCs, and we have accumulation of ILC1s. So we want to study what's the impact of this accumulation of type 1 ILCs in the gut of inflammatory bowel disease patients. To do that, we start by doing some mouse studies where we establish mouse intestinal organoids, and we co-culture with ILC1s isolated from the mouse intestine. As you can see here already in the pictures, the co-cultures of organoids with ILC1s, you can see an increased number of crypts in these co-cultures, and this correlates with increased intestinal epithelial proliferation in these cells. So we knew that ILC1s can produce a cytokine called interferon gamma, so we want to know if those ILC1 driven increased proliferation was by interferon gamma. So we add interferon gamma to the organoids and we didn't see it. So this was indicating that was a different pathway driving this proliferation. So to test that, we cultured the organoids alone or in the presence of ILC1s, and then at the time we just did bulk RNA-seq on intestinal epithelial cells. Many pathways came changed, and one of the pathways that was indicated it was likely changed in this condition was driven by TGF-beta. So this is another cytokine. So we want to see that was not linked to innate lymphoid cells at the moment when we are doing these studies. And so we want to see what's happening. So we had the organoids alone and the organoids with ILC1s, and we saw increased levels of this cytokine TGF-beta in the supernatant of organoids co-cultures with ILC1s, indicating that ILC1s were likely expressing TGF-beta. In addition, in these conditions, the intestinal epithelial cells were not expressing TGF-beta, but they were expressing the receptor for it, means that they could act and be influenced by the presence of TGF-beta. So to look if this was affecting the number of crypts in the organoids, we had again organoids alone, organoids with ILC1, so increased number of crypts, and then if we block TGF-beta with a TGF-beta blocking antibody, there's decreased epithelial proliferation. So in conclusion, ILC1s were driving increased epithelial proliferation through TGF-beta. Another pathway that was changed was linked to CD44. So CD44 is a molecule with many different functions and also many different isoforms. And the variant 6 isoform expression has been associated with IBD and intestinal cancer initiation. And this was actually the isoform that was upregulated in our cultures when the organoids were cultured with the presence of ILC1. And then again, because we knew there was a role of TGF-beta in this pathway, then we blocked TGF-beta and this increased expression of CD44V6 goes down. And it goes up again if we just add TGF-beta to the organoid cultures. So ILC1 and TGF-beta drive increased CD44V6 expression. So we were very excited that we had found a new role for ILC1s and how they interact with the intestinal epithelial cells. But we wanted to know, this was done in mouse models or mouse organoids. We want to know if this was relevant in the human setting. So to do that, we use iPSC-derived intestinal organoids and then we spoke about in the beginning of this talk, this iPSC-derived intestinal organoids, in addition to the epithelial layer, they are surrounded by mesenchymal cells, and then that end up being very important for our findings. And they were co-cultured with innate lymphoid cells that we isolated from patients' biopsies. So when we do that, we had the organoids alone and organoids with ILC1s patients in remission and organoids with ILC1s isolated from inflamed tissue of IBD patients. You can see there is increased expression of CD44V6 both in the epithelium and in the mesenchymal layer when inflamed ILCs are present. At the time we look at these cells were able to express TGF-beta, and they expressed it, and now we have a new story coming up very soon, how ILCs are actually able to express and activate TGF-beta, and how that's important in a lot of interactions with the epithelium. TGF-beta is known for its roles in modulating matrix remodeling, so we want to check if ILC1s were driving matrix remodeling in this context, since they were able to express TGF-beta. So for that, we grew the organoids in fully synthetic hydrogels developed by the Gentleman Lab, and then we add ILCs to these organoids and see what was their impact in the matrix remodeling. We use atomic force microscopy and for that we apply a bead functionalizing cantilever to the hydrogels and then measure the Young's modulus. And by doing that, we saw that they were both areas of stiffness and areas of softening when the organoids were cultured in the presence of ILC1s. This indicated that was likely matrix production and matrix degradation happening. So to look at matrix degradation, then we did microparticle rheology, where we add these little fluorescent beads into the hydrogels where the organoids were with and without ILC1s, and then measure the Brownian motion, so the motion of these beads in the hydrogel, and more motion indicates that there was degradation. So when we look at organoids alone, or organoids with ILC1s at day 7, you can see the bead motion increases, and the same at day 14. This indicates that there was matrix degradation in the presence of ILC1s. Now we know that these ILCs can express matrix metalloproteinases, and that's likely why there was matrix degradation in the presence of these cells. To look at matrix production, we look at the expression of some extracellular matrix proteins like fibronectin, and you can see its increase in the presence of ILC1s in the organoids. And this is shown very clear in this picture, where you have organoids alone or organoids ILC1s, and you can see this huge layer of fibronectin and fermenting surrounding the organoids. So in summary, ILC1s from inflamed tissue promote fibronectin deposition and matrix remodeling. So we have shown that human intestinal ILC1s can induce both matrix remodeling, degradation and stiffness, and also CD44V6 expression in the intestinal epithelium and in the mesenchymal layer. We were a bit surprised by these results because we set up these experiments thinking we were going to study the role of ILC1s in inflammation and how might they drive inflammation in IBD, and we ended up looking at cancer and fibrosis. We went back to our bulk seq RNA-seq of the intestinal epithelial cells cultured in the presence of ILC1s or alone, and the pathways that were changed in the presence of ILC1s were actually growth of tumor and fibrosis. So this is in line with our results and led us to propose that this excess of ILC1s in the inflamed tissue of IBD patients might be driving IBD main sequelae that are actually cancer and fibrosis. And fibrosis is the main reason that these patients require life-altering surgery. So for the next story, we were back to looking at what happens of ILCs in the intestine of IBD patients, and when I show you this graph before, I called attention for this increase of type 1 ILCs in inflamed tissue of IBD patients. And this comes to the expense of the reduction of these NKP44 or NCR positive ILC3s in IBD. So ILC3s are very important for the maintenance of epithelial barrier. And that's why we want to study what regulates this ILC phenotype in the intestine, and what do intestinal epithelial cells and how do intestinal epithelial cells and ILC3s interact. So to do that, we co-culture ILC3s isolated from the mouse intestine with small intestinal organoids, And we had done before where we have had the organoids alone or organoids with ILC3s and then bulk seek the epithelial cells to see what happened. And when we did that, we could see that in the presence of ILC3s, there was an increase in this goblet cell signature. So goblet cells are secretory cells and barrier protective cells. They are present along the cryptphilus axis. They produce mucus, and there is a mucus layer in the small intestine and a double mucus layer in the colon, and they are known to be MUC2 positive. There is an issue in studying these cells in the tissue versus the organoids is that the proportion of goblet cells present in the organoids is quite smaller in comparison to the one present in the tissue. But organoids are an amazing tool that you can manipulate, and there are protocols now to induce the expression of different differentiated cell types in the organoids of your interest, so you can induce higher expression of goblet cells, or tuft cells, or pannet cells using established protocol. So we did that, and we tested, and using this established protocol, we could increase the number of goblet cells in the organoids, or the proportion. So we had these goblet cells enriched organoids and our normal organoids, and we want to know if they were able to differentiately modulate the ILC3 phenotype. So to do that, we co-culture the organoids with ILC3s and then analyze them by flow cytometry, and you can see that the proportion of NKp46 ILC3s is enriched in goblet cells enriched organoids. And more important, the proportion of these cells that produce the protective cytokine IL-22 were also increased in the goblet cells enriched organoids. Goblet cells induce IL-22 production by ILC3s, as we just see, so we want to see if this impacts the intestinal epithelium. IL-22 targets different genes, as for example, the antimicrobial peptide REG3 gamma. To test this, we did our co-cultures on organoids alone, on organoids enriched with goblet cells with and without ILC3s. So when do that, the organoids alone, both normal and enriched with goblet cells, express very low levels of REG3 gamma. When we add ILC3s, these levels increase, but these levels are even higher in the goblet cells enriched organoids. This means that goblet cells induce ILC3s and expression of IL-22 has a feedback on the intestinal epithelium. So we want to figure out what was the mechanism. And as I've mentioned in the beginning, we have a way to know if this mechanism was cell-cell contact dependent or not. So we did the same experiments now in trans wells where we separate the organoids from the ILCs, and we stopped seeing this increased expression of NCR positive ILC3s. So we knew it was a contact dependent mechanism. You start to think maybe it could be notch dependent. So notch ligands DL1 and DL4 are expressed by the signal sending cell that then bind to notch on the receiving cell that can then translocate to the nucleus and has a lot of effects in target genes. So we want to know what were the source of notch ligands in the intestine and look at published literature and data sets, and we can see that the goblet cells are a good source of DL1 and DL4. So we start to hypothesize that maybe goblet cells were mediating this mechanism by DL1 and DL4 ligands. So to test this, we have our organoids, and we transfect with siRNA to knock down DL1 and DL4. Then we enrich them for goblet cells, and then we culture them with ILC3s. And this sounds like a very simple experiment. It took almost a year to set up. It's actually technically very complicated. But it was worth it, because when we did that, and then we look at the levels of IL-22, so the protective cytokine expressed by ILC3s, they are down when we knock down DL1 and DL4. So goblet cells are a source of notch ligands for ILC3s, and they drive their functions. So then the reviewers asked us if these cells were actually interacting. So we used these M-Sherry MUC2 organoids provided by the collaborator, and then we co-culture with orgamety positive ILC3s that were labeled with the green fluorescent protein. So I'm calling your attention for this green cell, so this ILC3 that is going to move, contact the red cell. So a goblet cell is going to become yellow, and then it's going to move out again. So let's see this. So you can see the green cell contacting the goblet cells and moving out again. So ILC3s can move in the intestinal epithelium and they can contact goblet cells, and that through notch ligands they might be driving this mechanism. So we show that there is this bidirectional signaling between ILC3s and intestinal epithelial cells, where goblet cells provide notch ligands DL1 and DL4 that drives increased NCR expression of ILC3s, increased IL-22 production that in turn drive, for example, production of antimicrobial peptides by intestinal epithelial cells. So for this final story, we go back to the beginning. And so the beginning is how ILCs develop. So ILCs can differentiate from precursor populations. These are called ILCPs or ILC precursors. And these precursor populations have been found in several tissues, organs. What led us to hypothesize that there is us and others that what happened is this tissue ILC poiesis, what means that the differentiation or the development of ILCs can occur at the tissue level and not in a central organ. And to support this hypothesis, the evidence that the ILC subset distribution and phenotype is different between different tissues. That's very clear in mouse models, where in the small intestine, the majority of ILCs are ILC3s, while in the lung, they are ILC2s, and then even the phenotype of these cells is quite different, especially ILC2s between the lung and the small intestine. So we want to know if the intestine itself were able to support ILC maturation, and to do this, we isolate ILC precursors from the mouse bone marrow and we co-culture them with small intestine organoids. And we did that drove the differentiation of all ILC subsets just by culturing ILC precursors in organoids. More important than having all ILC subsets is their function. So we compare the capacity of for example, expression of cytokines of these ILCs generating organoids with ILCs isolated directly from the mouse intestine, and for example ILC1 capacity to produce interferon gamma, ILC2 capacity to produce IL-5, and ILC2 capacity to produce IL-22 is equal to primary ILCs. We also look at the subset distribution of these cells, and in the intestine, I had mentioned before, the majority of ILCs are ILC3s or group 3 ILCs, and the same is what happened in our intestinal organoids. So we're curious to know if this capacity to drive ILC differentiation and maturation was unique to the intestine or other organs have it. To test that, we established lung organoids and co-culture again with bone marrow, with ILC precursors isolated from the bone marrow. And again, we were able to see that these organoids were able to drive the development of all ILC subsets. And very interesting, the subset distribution was different in the lung organoids, where we had a bigger proportion of group 2 ILCs or ILC2s. And in addition to and this is exactly what happens in the tissues that were in the lungs, there are more ILC2s, while in the small intestine, there are more ILC3s. So the tissue itself was driving this subset-specific distribution. More than the subset-specific distribution was driving also different phenotypes of the cells that were developing. So in the intestine, normally ILC2s express this marker called KLRG1, while in the lung they tend to express IL-33 receptor that is ILST2. And that's exactly what happened to our ILCs that were developing organoids. So the ILCs that develop on intestinal organoids express KLRG1, while ILC2s that develop the lung organoids express ST2. So the organoid identity was driving these tissue-specific patterns of ILC development. So it was very interesting that we were able to drive and understand how the epithelium drives ILC development, but these initial studies were done in mouse, and to study ILC development in mouse, we can actually use mouse models. What is really hard is to study ILC development in humans. The ILC precursors in humans have been identified a few years ago, and they are really easy accessible from the blood. So then we co-culture them with human iPSC driving intestinal organoids. And as I have mentioned a couple of times by this talk, these iPSC driving intestinal organoids have the epithelial layer, but also have a stromal layer of fibroblasts surrounding the epithelium. And this was very interesting because we thought we can then use this model to understand what was the role of the epithelium versus the role of the mesenchyme driving ILC development. And the reason this is interesting is because ILCs, like other lymphocytes, have been for years grown by immunologists just in this monolayer of OP9 delta-like 1 stroma cells. So we thought maybe having the mesenchymal cells was going to be favorable for the development of ILCs. So to do that, we had our organoids alone that have this EPCAM or epithelial cells present. You can see them here in green in this FACS plot. These middle cells in red, these 27% are our fibroblasts, and then we deplete these fibroblasts, so we went from 27 to 3%, and co-cultured these organoids with depleted fibroblasts with the ILC precursors. Then we had ILC precursors just with the fibroblasts, or ILC precursors alone. And we start these cultures as little as 500 ILC precursors per well, and after 2 days we have this massive expansion of the number of cells. So especially in our mesenchymal depleted organoids, we can have an average 750-fold expansion, so around 400,000 cells, or way more than from what we start our 500 ILC precursors. So ILC intestinal epithelial cells were able to promote this generation of innate lymphoid cells, and this was particularly happening when the fibroblasts were reduced or almost not present. And this is amazing because we are very used to work with innate lymphoid cells, and sometimes from 12 patient biopsies, we have in a good day like 3,000 cells to work, and now we have 100,000 of cells. So imagine the number of experiments we can do with this, the number of things we can actually figure out how human ILCs work. So now this is a very well technique used in my lab by many different projects where you just make a lot of ILCs, and then we can study their functions and interactions with others, and this protocol has now been adopted by other labs. So way more important than making a lot of cells is how these cells behave. So first, looking at the cells distribution, you have, again, because it's the gut, more ILC3s than other subsets, and then we extensively characterize these cells by flow cytometry, transcriptomic, and functional assays. And our ILC1s, and then were able to produce interferon gamma. The natural killer cells were cytotoxic, able to kill target cells. ILC2s, able to produce type 2 cytokines, and ILC3s, again, able to produce IL-17 and IL-22, and they were also able to present antigens as has been reported before. We did a single-cell RNA-seq in these ILCs, and we have, again, the different ILC subsets. And they express the transcription factors and gene signatures that were expected for each ILC subset. We also have a population of cells that has still a precursor signature that indicates that we further expand these cells in culture. Maybe we could increase the numbers that we get. Then we want to look at this tissue-specific imprinting given by the organoids. So we compare the ILC3s, that's our biggest population, with circulatory ILC3s, intestinal ILC3s, and lung ILC3s from published data sets. And you can see the cells have this circulatory signature as they come from blood precursors, but they already acquire a very strong intestinal signature just by being 2 weeks in culture with intestinal organoids. They definitely don't have a lung signature, so the organoid is imprinting this specific tissue-specific phenotype. We have done studies and we know that iPSC-derived lung organoids are also able to drive the development of ILCs. And we are now going to sequence these cells, and we expect to see is that they will have acquired a lung signature and won't have an intestinal one. So in summary, the epithelium supports the development of tissue-specific ILCs. We are able to generate all ILC subsets from the same culture under the same conditions. The cell's phenotype and functions are similar to their in vivo counterparts, and this expansion and differentiation is driven by epithelial cells. And the organoid itself imprints a tissue-specific phenotype. So ILCs generated in intestinal organoids have an intestinal phenotype, while ILCs generated in lung organoids have a lung phenotype. So now this has potential to be used as an alternative to animal studies, but because if you can make a lot of human ILCs, then we can use them for our studies instead of having to use the mouse ILCs, and also has the potential to be used for autologous cell therapies. So we are able to make ILCs with the phenotype of the organ that we want to target. So if you want to make, for example, protective ILC3s as a future therapy for IBD patients, we can make them with intestinal phenotype that drive for example, their migration to the intestine and their function and interactions with intestinal epithelium. Finally, I'd like to acknowledge all my lab has been involved in this work, especially people that I have mentioned throughout the talk, such as Geraldine Joette, Emily Reed, and Anis Penacerra. Isabel Coyales and Emma Croman also have been involved in the single-cell sequencing data that I have shown you. I'd like to acknowledge our collaborators that were essentially for the development of different stories and allow us to diversify and do interdisciplinary research. We couldn't do this without the support of the funders and also without the samples from the patients that allow us to do this very interesting work. Thank you very much for listening, and I look forward to answering your questions pretty soon.
Thank you very much, Joanna. And on to questions. So can you please provide a bit more detail about the main challenges in growing intestinal organoids specifically?
So it really depends from where you are growing, if it's from iPSC or human, so tissue-derived organoids. So I think the challenge is the variability you get from tissue-derived organoids, and sometimes are not able to establish organoids from all the patients, and iPSC also depends on the lines that you use. But in general, I can say it's a very robust and well-established protocol, probably the easiest organoids to grow, and I think it's quite easy to be adapted by groups that don't have experience in organoids.
Wonderful, thank you very much. And a follow-up question, how often do you see the generation of a necrotic core in intestinal organoids? Is it about average compared to other types of organoids, or is there anything unique?
I cannot comment on comparison to other organoids. So in like other organoids, they are closed if you think like a tennis ball, and every debris is produced by the inside, but it's completely fine. We passage them every week, and there's no issue with that.
Wonderful. Thank you very much, Joanna. Dr. Natalia Vergara is an assistant professor of ophthalmology at the University of Colorado School of Medicine. Her research team uses human stem cells to model retinal degenerative diseases and applies these models to translational research aimed at discovering and developing therapeutic drugs. Born in Argentina, Dr. Vergara earned her PhD in biology from Miami University. She then completed her postdoctoral research at Johns Hopkins School of Medicine, where she contributed to the development of the first light-responsive retinal organoid model derived from human-induced pluripotent stem cells. In 2017, she joined the faculty of the University of Colorado as part of CellSight, the Ocular Stem Cell and Regenerative Research Program at the Sue Anschutz-Rogers Eye Center. Her work on retinal organoids has been recognized with 3 awards from the National Eye Institute. Let's make sure our slides are up and running. And the floor is yours, Natalia. Thank you.
Good morning. My name is Natalia Vergara, and I'm an assistant professor at the Sue Anschutz-Rogers Eye Center in the University of Colorado School of Medicine, United States. Today, I'm going to talk to you about the work that we're doing here using retinal organoids to accelerate drug development for retinal degenerative diseases that cause vision loss or blindness. So the retina is a light-sensing tissue in the eye. It lines the back of the ocular globe, and it is an extension of the brain. So the retina is part of the central nervous system. And just like the central nervous system is composed of neurons and glia. And what is meaningful here for this talk is that as an extension of the brain, the retina lacks regenerative capacity, which means that any disease that is going to damage these retinal neurons is going to cause irreversible vision loss or even blindness. Retinal degenerative diseases affect millions of people worldwide, and they can be inherited, as in the case of retinitis pigmentosa, for example, which is a group of inherited retinal diseases that cause tunnel vision. And so the patient starts losing their peripheral vision, and the visual field becomes more and more constricted until they can go completely blind. But they can also be acquired, as in the case of age-related macular degeneration, which is one of the leading causes of blindness irreversible blindness in the world, and cause the loss of central vision. The problem with these diseases is that there's no current treatment to replace neuronal loss in the retina once they are damaged. And that causes an urgent need for innovation. We need to develop drugs that can prevent that neuronal loss before it happens. And if that is not possible, then we need to develop strategies to replace those retinal neurons, strategies that, by the way, don't exist yet. So a solution to this problem is the use of human stem cell-derived retinal organoids for therapeutic development, and that's what we're going to be covering today. What we are using are human-induced pluripotent stem cells. This is a very powerful technology, and so much so that in 2012, this technology won the Nobel Prize. And that was just 6 years after the first paper on this came out in 2006. And it is so important because it just revolutionized both biology and medicine, because it allows us to take cells from a donor, any donor, and almost any cell type. Originally, these were made from skin biopsies, but nowadays they're made from blood and even from urine. And those cells can be reprogrammed to become induced pluripotent stem cells that mimic embryonic stem cells in their capacity to proliferate and to differentiate into any cell type in the body. So now we can push the differentiation of those stem cells in 2 dimensions or even in 3 dimensions to form these tissues that we call organoids. And the question we and many others asked many years ago was whether these cells are able to generate retinas in vitro. And the answer is, of course, yes. Retinal organoids can be generated from human-induced pluripotent stem cells following a stepwise process. And as you can see here, we can generate it even by the hundreds in a petri dish. These are mini retinas that really mimic the structure of the retina, except that they're only about up to maybe 1.5 or 2 millimeters in diameter. But if you look at their histology, and this is a very busy slide, but just to give you an idea, these mini retinas in vitro are able to generate all the different cell types that you see in a normal human retina, including ganglion cells that are the ones that in vivo are making the connections between the retina and the brain, and photoreceptor cells, which are the cells that capture light, and those are your rods and cones, depending on whether you're looking at low intensity light or color vision. Now, very interestingly, these retinas that are growing in vitro are even able to respond to light stimulation. And that is something remarkable because it means that they have to develop all the machinery that is necessary for light detection and phototransduction. And they have been shown to even transduce a signal from the photoreceptors to the interneurons in the retina. The method that we used to generate these retinal organoids was published by Zhong et al. In 2014. And briefly, what happens here is that we lift the colonies of iPS cells and we grow them in suspension where they form embryo bodies. And then eventually we plate them again in attachment onto matrigel coated dishes, and they start spreading out and forming these 3-dimensional structures that we're able to see in a bright field microscope. And so we take tungsten needles and we cut around these structures to lift them up, and we grow them in suspension for the rest of the time. And when we do that, these structures fold onto themselves and form these retinal cups that are basically very young retinas that continue to mature over time to a point where they form this specialized structure. So what you can see here is a close magnification of an older retinal organoid, and those structures here are the photoreceptor outer segments, which indicate that those retinas are a lot more mature. Of course, that's not the only protocol to generate retinal organoids. I don't expect you to be able to read this, but here is the reference, and there's many other references. This is a review that compares different protocols that have been generated to make retinal organoids. But the bottom line is that all these protocols, there are slight differences, but in principle, they're all very, very similar. So how is it possible that we can generate retinas that look just like an embryonic retina from stem cells in vitro? And the reason is that they just recapitulate development. So far, even though people have tried other things, the only way at this moment that you can generate a retinal organoid is by allowing those iPS cells to develop just as they would in the embryo. So each of the steps that you see in normal retina development, from the formation of the eye field in the anterior neural tube to the invagination of the neural tube to form the optic vesicles, the invagination of those optic vesicles to form the optic cups, and then the cell type specification within that early retinal neuroepithelium to the maturation of all the different cell types. Each of those steps has a correlation in the retinal organoid generation protocol. And they not only mimic retina development in space, but also in time. And this is very meaningful for translational applications because it means that if you take, for example, a mouse iPS cell, you're going to be able to generate a retinal organoid in about 4 weeks because a mouse develops, for 3 weeks, and then after birth, the retina continues to develop for an extra week. So 4 weeks, you have a mouse retina. If you take human iPS cells, it's going to mimic the timing of the human embryo, and it's going to take about 6 months for these retinas to generate photoreceptor cells that are mature enough to be able to detect light. So that is a very interesting model that, as you can imagine, has a lot of really interesting applications. But what is it that the model doesn't have? What are the things that we normally don't see in papers or even in talks? Or the dirty little secret. So one of the things the model doesn't have is a retinal pigmented epithelium juxtaposed to the neural retina. So the pigment epithelium is a layer of cells that basically sits behind the photoreceptor layer in the retina, and it's very important for photoreceptor function and survival in vivo. So age-related macular degeneration, for example, one of the diseases that I mentioned before, starts with the degeneration of the retinal pigment epithelium, and that causes the degeneration of the photoreceptors that lie adjacent to that. But in retinal organoids, we don't form that pigmented epithelium. Well, we do form it, but not juxtaposed to the retina. We form it, and it tends to cluster at what will be equivalent to the anterior portion of that retina, so in the wrong location. And we can talk later about the reasons for that, but they're mostly mechanical. This is relevant because if you want to model a disease like macular degeneration that depends on the interaction between those cell types, you're not going to be able to model that perfectly well with a retinal organoid. You can, however, grow the pigmented epithelium as a monolayer separately and then grow the retinal organoids and then juxtapose those artificially after you have grown the tissues. The other thing you're not going to find in retinal organoids are blood vessels, because the whole idea of generating these organoids is pushing the iPS cells to become neuroectoderm. And so the retina and the brain are neuroectodermal derivatives, and those are the only types of tissues that are going to be formed from this protocol. Blood vessels, therefore, are not formed. And so that means that if you are trying to mimic a disease that has a big component of vascular compromise. So, for example, diabetic retinopathy, you're not going to be able to recapitulate the interactions between the vasculature and the retina in this kind of model. You can, however, use the model and take advantage of that lack of interaction to explore more targeted mechanisms. For example, you can expose retinal organoids to high glucose levels or to different kinds of cytokines, and you can test things one at a time without having all the confounding factors that you would have in a more complex system or in an animal model. The other thing that doesn't form in retinal organoids is an optic stalk. The optic stalk is what connects the optic cap to the neural tube in the early embryo. And later on, it forms the canal through which the axons of those retinal neurons exit the eye and connect to the brain, basically forming the optic nerve. What happens here is that one, you do not have a brain, and two, you do not form an optic stalk. So those retinal neurons will form axons, but those axons are going to bundle in the cavity inside the organoid instead of exiting to form an optic nerve. That means that if you're studying, for example, an optic neuropathy, a disease or a condition or trauma that would affect the optic nerve, you're not going to be able to model that easily in retinal organoids. And the other thing that you don't have here are Mueller glia. Microglia. Microglia are the macrophages of the central nervous system. And they are yolk sac derivatives. They're not neuroectodermal, so they're not going to form here. You do have Mueller glia because that is formed from the same retinal precursor that forms all the retinal neurons. But microglia are not present. And so several groups are now trying to generate co-cultures of retinal organoids with microglia to see if they can recapitulate those interactions. The other thing that is a little bit vague at this moment is whether these retinal organoids can have astrocytes. Astrocytes are neuroectodermal derivatives, but during development, they are not formed from the same neuroepithelium that forms a neural retina, but rather invade the neural retina from the neural tube later on in development. And there are some RNA sequencing studies that have detected markers of astrocytes, but there aren't any histological studies that show where those cells would be or whether they would even be in the correct location. So that is a little bit up in the air at this moment. So in conclusion for this part, retinal organoids constitute a simplified system. They lack a lot of the associated structures, but they are able to recapitulate all the major events in retinal development in both a spatial and temporal manner, which is something that needs to be taken into account when we're trying to use these retinal organoids as models. One, because as I told you before, we need to understand that it's going to take a really long time to generate these models. And two, because depending on the time that you use these retinal organoids, the composition is going to be different and the level of maturation is going to be different. And that is something that needs to be taken into account in the experimental design. However, these retinal organoids are able to achieve advanced levels of maturation, including the ability to respond to light. And single-cell RNA sequencing studies have shown that when you look at retinal organoids that are much older, let's say older than about 200 days, they tend to cluster more with the transcriptomic profile of adult retinas than embryonic retinas at that point. So a model doesn't need to be perfect. Actually, no model is perfect, not even the animal models that are commonly used in research. The model just needs to be adequate to answer the question. And sometimes having a simplified system can be advantageous because it allows you the opportunity to test single variables at a time without a lot of confounding factors. So now we can use these retinal organoids for translational applications. So we can think about generating iPS cells from a patient, making these 3D retinal tissues and using them, for example, for transplantation, for cell replacement. And that is something that is being developed at this moment. But, the point of this talk is that you can use these retinal organoids to model retinal degenerative diseases so that you can use them for drug development to generate treatments for these diseases that right now lack effective therapies. And that is the goal of CellSight, which is the Ocular Stem Cell and Regeneration Research Program in the Department of Ophthalmology here at the University of Colorado. And CellSight is tackling all these different problems, but in particular today we're going to talk about the applications to drug development, and that can encompass, for example, using retinal organoids for screening drugs and validating their efficacy or potential toxicity, for testing gene therapy strategies or other delivery methods, such as, for example, nano delivery strategies. Now, is there a real need to use retinal organoids in drug development? Of course, and the answer is yes. So the reason for that is that there's many reports that say that even up to 90% of clinical trials fail. And we're talking specifically about retina here. But in many cases, and in many reports, part of what is used as an explanation, a culprit for that, or at least part of it, is the poor validity of preclinical models used in drug development. We're talking about 2D cultures, tissues, animal models. And therefore, institutions such as the National Institutes of Health in the United States or the Food and Drug Administration are now moving towards prioritizing human-based technologies for drug development. So can we use a retinal organoids to do high-throughput screening assays? Absolutely not. Not at this moment, at least. If we go by the definition of high-throughput screening. So normally high-throughput screening, we're talking about hundreds of thousands of compounds per day. That is not something viable in normal scenarios for retinal organoids because of the investment of time and the expense of generating that many organoids for a screen. But retinal organoids do have a place in drug development. So the idea here is that you could use simplified models like biochemical assays or 2D cultures to do these high-throughput screenings where you test hundreds of thousands of compounds. And then once you identify those hits, you can use the retinal organoids for hit refinement and optimization in a low to mid throughput. So you can test, as I said before, drug efficacy, toxicity, in specific effects within the retina, and refine those candidates and combine the retinal organoid models with those animal models. Can we get rid of the animal models? No, not at this point, or not with the technology that we have at this time, right? We still need to make sure that the drugs are not going to have toxic effect in other organs before we can go into a patient. And now I'm going to give you just a couple of examples of how in my lab we have used these retinal organoids for drug validation purposes. The first example I'm going to tell you about is very exciting, and it's the use of retinal organoids in personalized medicine for rare diseases. So we have a patient of Dr. Emily McCord here at the Children's Hospital in Colorado. And this little girl has a disease called PCARP, posterior column ataxia with retinitis pigmentosa. This is a rare autosomal recessive disease that is caused by mutations in a gene called FLBCR1, which is a choline transporter. And this causes neuropathy, but also photoreceptor degeneration, which means progressive blindness. And so a large team of people led by Dr. Timothy Yu at Boston Children's Hospital identified an intronic pseudogene insertion in this patient that resulted in an aberrant splicing that introduced an early stop codon. And so they developed an antisense oligonucleotide treatment specifically for this patient in order to restore the normal splicing. Now, how do we know if this is going to work in the patient's retina? And that's where the retinal organoids come in. So the wonderful Cami Howell in my lab and Dr. Boxon Sao in Dr. Yu's lab in Boston generated iPS cells from this patient and generated retinal organoids from the iPS cell. So what you can see here is just retinal organoids generated from a control line and from the patient, and a general validation of the structure of those organoids just to show that they form all the different cell types that you find in the retina. But the more meaningful part is that they showed by different methods that these antisense oligonucleotides were able to penetrate the retinal organoids. And once they did that, they showed in a dose response manner that they were able to reduce the missplicing of the transcript. So the red here is the aberrant splicing, and to restore the normal transcript, which you can see here in green. And this is very exciting. And so much so that after not only this, but also after doing testing in animal models to evaluate the potential toxicity, and establish safety and all of that, we were able to get an IND designation and permission from the Food and Drug Administration to start a clinical trial with an N of 1. And Dr. Mark Matthias here in our department started doing intravitreal injections of this treatment for this patient. This is very meaningful because let's think about this. This is a progressive degenerative disease that does not have a cure. And if we inject this antisense oligonucleotide therapy in this patient, we will have to wait years to see if the degeneration of this photoreceptor progresses or not, because this is not going to restore the cells that were lost already. What it's going to do is prevent further vision loss. And because we only have an N of 1 in this clinical trial, we're not able to compare to see if, this degeneration has slowed down. So the only way we have to check whether this treatment has any potential to work in this patient is by doing this work with the retinal organoids. And that is the power that it can bring to personalized medicine. And I'm going to give you just a second example here about using retinal organoids to evaluate peptide-based treatments for photoreceptor degenerations. This is another case of retinitis pigmentosa. And this work is about evaluating a PDF, pigment epithelium-derived factor, derived peptide called H105A as a potential neuroprotective therapy for photoreceptor degenerations. And this is a work in collaboration with Dr. Patricia Becerra and Alejandra Bernardo Colon at the National Eye Institute and Dr. Valeria Marigo at the University of Modena. So what they showed using a mouse model of photoreceptor degeneration and delivering this peptide in the form of eye drops was that, and you can focus on this part here, this is a staining in live animals for cell death. And we're looking at a fundus of the eye, looking basically at the retina. So red means more cell death in the retina. And you can see that in the vehicle control, we have a high amount of cell death at 21 days of age. And when the mouse has been treated with H105A peptide, that level of cell death is really, really decreased. And you can see that here in the quantification. And that is very different from what you see if you use an inactive peptide that does not have an effect, which is what you see here. And that was just one of the measurements. Of course, there was a lot more than in this paper, but for example, what you can see here is that the H105A peptide was able to preserve the thickness of the photoreceptor layer in these retinas in a manner that is more or less similar to control conditions. Whereas if you don't do this treatment, you see a significant reduction in the outer nuclear layer, which basically means a reduction in the number of photoreceptors. And we also obtained similar results in a different mouse model, a rhodopsin mutant, and also with a different form of delivery. So using, for example, a gene therapy based on an AAV2 vector. So basically, this peptide is able to protect photoreceptor cells in mice, but can it protect photoreceptor cells in a human retina? And that is what we tried. So the wonderful Dr. Shama Parwin, who's a postdoctoral fellow in my lab, developed a model using these retinal organoids where she exposed the organoids to cigarette smoke extract. And when she did that, she did a thorough characterization of this model. But, among that, what she found is this is tunnel staining for cell death. So what you're seeing here is a lot more cell death happening when you have exposure to cigarette smoke extract compared to control conditions. And you can see the quantification over here. And she also developed several techniques that you can use in live organoids that are very fast, quantitative, and reproducible and can be done using a plate reader. And so one of those techniques, for example, is staining the retinal organoids with dihydroethidium for measuring reactive oxygen species. And you can really see here, this is just a positive control, and you can see the DMSO control here. And when you expose the retinal organoids to cigarette smoke extract, you really increase the levels of oxidative stress in those organoids. And so we use that model to test this H105 peptide. And so what you are seeing here are these fluorescent-based live organoid assays. And this is PSView. So basically what this does is it measures cell death in those retinas. And so this bar here is your cigarette smoke extract. And these here are the treatment. And so you can see that there's a significant reduction in cell death by these metrics when you treat with H105A compared to cigarette smoke treatment alone. And that is reproducible with other techniques. So we saw basically the same idea. This is cigarette smoke extract, and this is that in addition to H105A, and you can see a significant decrease in the amount of cell death. And that was even validated by histology in sections measuring tunnel. So the takeaway from this is that the PDF-derived peptide H105A has a good penetration into the retina, which I didn't really show you here, but it can be administered as eye drops, and it can protect photoreceptors from degeneration, both in a mouse model and in a human retinal organoid model. And that could constitute a potential gene agnostic therapy for retinitis pigmentosa, which is as I told you before, it's an umbrella of diseases. There's many different gene mutations that can lead to retinitis pigmentosa. And the problem with that is that one, is very difficult to develop specific treatments for every patient and for every mutation. The number of patients for each mutation is not very large. And on top of that, there's a large percentage of patients with retinitis pigmentosa for whom we cannot identify the disease-causing mutation. So developing a gene agnostic therapy is really something that is a high priority. And this is an example of how retinal organoid models can complement animal studies for therapeutic development. So in summary, here I've shown you how retinal organoids can be used both for personalized medicine approaches and to test drug efficacy and drug toxicity and validate them so that then we can move towards preclinical studies to take those therapies back to patients. And with that, I'd like to thank my lab and all the collaborators. It really takes a village to do all these kind of studies and all our funding sources. And thank you very much for your attention.
Thank you very much, Natalia. We have a couple of questions. Our first question is, how far out from preclinical studies would you say this kind of work is?
Well, it depends on what you're talking about, right? So, as I mentioned before, this particular personalized medicine approach that we use is already being administered to a patient. The use of organoids in general for preclinical research is not just something that is being done, is something that is going to be required, at least in the US, moving forward. And so I think the key here is to make sure that the technologies are optimized for quantitative studies, right? And this is something that I've shown in the presentation, and that we spent many years optimizing just to make sure that we can deal with the variability of the system and that we can have fast and quantitative outcome measures that are applicable to this kind of applications. If you're talking about H105A in particular, I understand that our collaborators at the NEI are ready to start preclinical studies for this particular product.
Wonderful. Thank you very much, Natalia. Welcome back, everyone. I'd like to introduce our final speaker. Dr. Casey Ronaldson-Bouchard is co-founder and chief scientific officer at Link Biosystems, aiming to democratize precision medicine with scalable bioreactors and platforms for modeling disease. As an associate research scientist in Columbia University's Department of Biomedical Engineering, she crafts multi-organ tissue models from pluripotent stem cells to build patient-specific avatars with inter-organ interactions. Before Link, she co-founded Tara Biosystems, a human heart-on-a-chip startup later acquired by Velo Health, and her work has appeared in Nature, Cell, and Nature BME. Let's make sure your slides are up and running. Casey, can you please share your screen?
Yes. How does that look?
It's loading. I'll let you know in a moment. Okay, great. The floor is yours, Casey.
Awesome. I'm Casey Ronaldson-Bouchard from Columbia University, and I'd like to talk to you about how we use patient-derived organoid models and engineered tissues and kind of piece them together to make a systemic model of different human diseases. So as I'm sure you guys already know, and has been said a lot today, drug testing overall just really isn't quite predictive. So you've got these 2D assays where cells are flat on a petri dish. You can make them a bit more physiologically relevant by culturing them in 3D or in organs on a chip. And then you've got the animal model, which is a whole system. It's systemic, but it's not human. And so piecing these together, you end up having about 10 years to get a drug to market. It's costing billions of dollars and it's having very low success rates. So 8 out of 9 drugs are failing. And then when you do get a drug to market, it still really fails to work for most people. So on the right, you'll see those are the top 10 blockbuster drugs. And at best, they work for a fourth of patients. So even when you are getting drugs to market, they're really not working for everyone, or they're working for barely anyone. And so part of the problem is that we're not really seeing the whole picture of human biology. And I think it's akin to this old tale about how these blind men are walking through and they come across an elephant, which I know is very likely, of course. So they're all feeling a different part of the elephant. And then they're talking later by a campfire and they're describing what they saw and what their conclusion was. And they come up to these drastically different conclusions about what they saw. You know, one saying it's a spear, it's a snake, it was a tree, a wall. You know, they didn't have the whole picture. And so they came to these completely different conclusions. And you can say that we see that in drug discovery or medicine in general, where you're only getting pieces of the story. You'll have like a liver toxicity assay over here, a mechanistic screen there, large primate studies. So you have pieces, but they don't necessarily tell you the whole picture. And, you know, not even a full clinical trial, or not until you get to the full clinical trial do you even start to get that. And so our solution is just to put, more puzzle pieces in, and so you can get more of a clear view, definitely not solving the view, but to have these diseases in a dish or human tissues in a dish that represent the different organs. And then you can connect them together. And then they also take in that patient diversity. So we're trying to, make systems that can develop drugs for all. And so how do we get there? So we're merging organoid biology with organs on a chip, and we don't necessarily have a preference for any of them. We mostly like to engineer the complexity needed to answer our biological question. So definitely our model systems might change a lot, and we like that built-in flexibility so that we can really focus on the question at hand and not necessarily be tied to the technology. So organoids are great. They give self-organization. The cells really know what to do. We always say that put the cells in the right environment and you don't even have to engineer them too much. They ultimately know what to do. So they'll drive their own developmental programs and structure right on their own. And then you can take that a step further and make more engineered tissues. And so here you're adding in a bit more control and function. And then organs on a chip or microphysiological systems where you have fluidics let you either kind of connect multiple organoids or engineered tissues, or just take one of those systems and add that element of perfusion, which helps to mature the tissue, keep it more alive, and then also lets you deliver mechanical cues or have immune cells circulating, things like that. And so just a little bit about engineered tissues and organs on a chip, since they're a little bit different than organoids. And so the main goal here is to just define one minimal functional unit of the tissue and try to replicate that. And so it's mostly looking at functionality that you can measure. So for the heart, it's contracting. And so we tend to think of, a strip of heart muscle that can contract, and then engineering that functionality in, we electrically stimulate it so it can contract. In the lung organ on a chip models, it breathes. So you have that mechanical stretch. So that's replicated. If you're looking at like a sarcoma model, you would put the cancer in a bone and have that mechanical loading. And so it's really trying to recreate the function that you see in vivo. And so here's a case study where we took organoids and engineered tissues, and then we combined them within a multiple organ on a chip platform. As specifically, we're looking at making a model of systemic disease, notably doxorubicin-induced cardiomyopathy. So our design constraints were that it be patient-specific, but still have high biological fidelity, have multiple tissues, but they're still connected together, and also still mature, even though they're all connected. And then we wanted to look at the system over longer periods of time. So we're getting readouts, not just one snapshot. And then we also wanted to make it modular and reconfigurable so you can add on complexity as needed. So this could be kind of a workhorse for, any systemic model you wanted to answer within the lab. So just a quick case study on why tissue maturation is really critical. So earlier it was mentioned that iPS organoids have that more fetal-like phenotype versus those you're getting from primary cells or straight from the patient. And this is quite true, but especially working with the heart, we need to rely on iPS cells because we're not necessarily taking tissue from anyone's heart. So we get those iPS cells, and then we make them into cardiomyocytes, which are the main cells of the heart, and they beat. They really mimic the heart. However, they don't necessarily have that biological fidelity because they're really fetal-like. So for adult cardiomyocytes, if you expose them to the drug isoproterenol, it'll increase in force. You'll see this increase in force with dose, and also this increase in calcium and beat frequency. And then when we do that with our iPS cardiomyocytes, you don't see that. It's completely blunted response. And so we don't have that maturity. However, in your body, your heart takes quite a bit of time to develop. Even when you're born, it's still very neonatal. And so we wanted to figure out how could we basically accelerate maturation. So what we kind of came after many years of studies, we discovered that taking early stage cardiomyocytes, so as they're going from iPS cells and starting to commit to that cardiac lineage, right when they're committed, we take those cells and use them then. Because if we let them commit too long, then they're not necessarily as plastic. And so they're not going to respond to the signals that we're giving them afterwards. So we take these earlier stage cardiomyocytes, put them in a 3D tissue with supporting cells, and then the heart beats in response to this electrical signal. So we also added electrical signals, and we would tell it to beat. And every day we would increase how fast it beats by 0.33 hertz. And so it always had to adapt and work harder and harder. And so it would kind of accelerate this maturation. And so this training regimen was what we used to mature our tissues. And so now we have our mature tissues. You see that you have a lot of mitochondria, aligned sarcomere, T-tubules, all the hallmarks of a more mature tissue. And then when you expose it to isoproterenol, you're getting that drug response that you should see where you're having that increased force and increase in calcium. And so now that we've worked so hard to make just one engineered tissue more mature and adult-like, how do we put that into a system with other engineered tissues and not sacrifice all that biology that we worked on helping to engineer? So that's something that we keep trying to balance. And a big component of that is the media that cells are in. And so we wanted to let them connect to each other and share signals without making them all sit in the same media bath. And so we kind of took the strategy of, looking at the human body and how do organs in your body talk to each other, and they talk to each other through your vasculature. But it's quite hard to vascularize micro and macro vasculature within every tissue. And so we tried to simplify it, and we just flattened the tissue, the vasculature. And so underneath each well, we have a custom transwell insert that we put vascular cells and supporting parasites on, and that gives you enough of a barrier. And so then you can just drop in your engineered tissue or organoid on top, and it's in its own media. You can add your own cues, so it lets you connect them while still having their own place. And so this is a bit what it looks like, our multi-tissue platform. And so it just snaps all together. You drop in your vasculature, and then you can just drop in any tissue on the top and fill it up with its media. And then that's the bottom. So the perfusion loop kind of runs underneath, and you can put immune cells in there and keep everything quite happy. And then What's nice, what we liked about our system, so with a lot of organ on a chip or microfluidic systems, they're closed and bubbles become quite a problem. And so everything's open, so you can sample really easy, and bubbles will just float to the top. So we kind of have these built-in bubble traps. So overall, the platform lets you have tissue-specific niches plus perfusion through the vasculature. So to put the pieces all together, first we engineered the tissue separately, and then we transferred them to the platform. And so we took this approach instead of engineering everything on the platform, because if one tissue fails, then the whole chip would be bad, right? And there's also different timelines for each tissue for maturation. And so it was much easier just to, do that separately, so that we could really have quality control built in for each tissue before connecting them. And so how we engineer them, so the heart I went over, but you see we put it in this intensity training regimen. It can capture electrically much better at a lower voltage, and then has this increased force with increased calcium that you would expect. For liver organoids, we'll take hepatocytes, and then we always add supporting cells to our tissues because it just helps keep them alive and in 3D. And so they'll self-assemble into these organoids that will then put in a bigger fibrin hydrogel. And you'll see that tissues, especially hepatocytes, when they're cultured in 3D versus 2D, they're able to sustain albumin production over time. And they very quickly lose that in 2D. The bone tissue, we take a piece of bovine bone, we mill it to a certain shape, decalcify it, and then we'll put the human cells back in. So we put in osteoblasts, which make bone, and then osteoclasts, which eat away at the bone. And so you'll see we can have these changes in bone volume over time. And then for skin, it's made in a typical transwell approach, where you make your dermal layer with collagen and your fibroblasts. And then once that forms, put keratinocytes on top. And then once you expose it to an air-liquid interface, the cells know what to do. So they stratify, and then you've got a nice skin barrier function. The vasculature, we have these transwells. We played around with the sizes, but ultimately settled with 20 microns because we wanted to have as much cell-cell contact as possible and not have as much, artificial substrate in there. But 40 microns, we would still get holes in some spots. So 20 microns was kind of the sweet spot for us. But once we put the cells on there and expose them to shear by running that perfusion loop, they get a really nice vasculature barrier that has, a great resistance, electrical resistance, which shows the stability, and we can look at the transport of molecules across it. So looking at validating our platform and seeing, what can we actually use it for, we kind of broke it up into a couple of studies listed here. So let's go through them. So inter-organ communication. So how, are our tissues actually talking to each other? And so we started tracking a lot of cytokines, but that, became quite difficult to track back to a tissue because some of the cytokines overlap or could be from the vasculature. So we decided to make something that we would absolutely know came from a certain tissue. So we took our cardiac tissue, our iPS cardiomyocytes were labeled for their exosomes. Any exosome from the tissue was labeled with CD63 that was fluorescent. So any exosome that the cardiac tissue is making, we could track where it goes. And we know that it came from the cardiac tissue because they are our only cells that are engineered with this fluorescent exosome reporter. So you can see that the vasculature barriers uptake the exosomes. And then while the heart, of course, has the most, it is still going to those other tissues as well. So our tissues are able to talk to each other. We looked at whether we could culture this over longer periods of time and how important the vasculature barrier was. And so our interorgan approach is with that vasculature barrier, but then our mixed is just having a transwell. So kind of more of a common media approach where they're sharing media to talk to each other. And then just the tissues in isolation as a control. And we see that so having that vasculature barrier really lets you preserve the individual tissue niche over long term. So the heart striations are staying, you still have albumin producing cells in your liver, and you've got the skin epidermis really staying intact. Same for our endothelial barrier. So it stays over time. And then in our liver, we could see a little bit of a leakier barrier, which is kind of a specificity that liver endothelial cells take. So that was nice to see. And then with respect to immune cells that are circulating, when we have that vasculature barrier, the cytokines stay a bit more stable over time. So that interorgan having the vasculature separation lets us have tissue-specific function over the long term. So we're seeing, the contractility stays, albumin production, trap activity in the bone, and then our skin barrier functionality. So we looked over, we wanted to dig a bit deeper instead of just functionality. So we looked at global proteomics, and instead of looking at what the organs are expressing separately, we asked what are they, all of the highly expressed proteins, what do they share? And so they shouldn't really share all that much because they're quite different organs. So in the interorgan one, where there is that vasculature barrier, you'll see the things they share are really, universal cell pathways, the same in the isolated. They're just like metabolic pathways and things like that. But if we look into the mix, every tissue shows signs of gene ontology for skin morphogenesis, bone ossification. So even the heart is showing bone ossification or skin morphogenesis. And this is likely because of that shared media, where the media is really driving these different pathways, especially because these are IPS engineered tissues. So they're more plastic and a bit more fetal, so very sensitive to mixing of media. We also looked at how they compared to adult gene expression and the overlap, and they showed quite a bit of overlap. With the bone here, it's bone marrow, because there aren't really just typical bone in any of the databases. Then we zoomed in even closer and looked at what are the top highly expressed genes for each tissue. And there you'll see the interorgan platform is really great at maintaining what would be expected of adult tissue. And then if we look into the mixed tissue, what's going on, we took the heart, for example, here, you'll see this upregulation in the mix of more stem-like pathways. So it's almost dedifferentiating. And then we're losing all of those cardiac cellular components that we worked so hard to engineer in. So our Z-disc, our T-tubule, everything related to the heart cell beating, we're basically losing. And so now we wanted to look at, the model system keeps the tissues alive and happy for long periods of time. And so then we wanted to look at what different models could we do. So we looked at a systemic immune injury response. So we took the heart tissue out and we cryo-injured it, and then looked at immune infiltration. And sure enough, it went, right to the heart, especially when we have the vasculature. In our mixed model, it goes to all the tissues. You can also see that with the attachment of the monocytes underneath the barrier and our troponin response with injury. This is maintained over a longer period of time. And then we wanted to ask the question, do immune cells just love the heart? And we put a healthy heart tissue next to an injured heart tissue and repeated the experiment. And it's the injury that drives the immune response, which is what we had hoped to see. And so a lot of using the platform now that we've kind of validated is always asking the question, how complex is simple enough, and what complexity do we really need? And so, it's not often that we need all 4 tissues. And so trying to always brainstorm from the beginning, how simple can we make it? So that way it's cleaner and more reproducible. So looking at it for PK/PD, do we need all 4 tissues or 1 tissue? So we did PK/PD modeling, and you'll see that for doxorubicin, in the mixed, it goes right into the tissue and gets absorbed quite quickly. But in the interorgan, you have more of that slower release profile and conversion to doxorubicin, the metabolite, and our PK matched our experimental data. So that was quite nice to see that we're able to, establish this model to look at where drugs are and how they're being metabolized. Then we further looked at, drugs metabolized by the liver and how they affect the heart. So difidolide should slow the heart down, where epinephrine should speed it up. And you'll see that it does, in fact, do that. And it also, epinephrine is really quickly metabolized. So you see that response, rapidly, and then it disappears. Whereas difidolide, it's a slower response. We also looked into, so in our DoxTox model, we wanted to see how could we benchmark these this whole system to clinical relevance. And there was a pediatric study that came out looking at microRNA biomarkers. And MIR1 was what was indicated. And we showed that, that's also in our platform seen. And that's even in the one tissue model. So just of a heart injury, you don't necessarily need all of the 4 tissues. And then if we looked into all of the microRNAs that they found and did PCA plots, we'll see that they overlap it with the clinical results quite well. And then zooming even further, you can see that the specificity and sensitivity is much higher when we have this inter-organ platform. We also have used it quite a bit for cancer modeling, so specifically metastasis, and seeing if we can, really make different models for the whole metastatic cascade from, that initial intravasation and priming of downstream organs to colonization. And so looking at breast cancer, we see that the breast cancer cells metastasize to the heart, liver, bone, and lung, or to the bone, liver, and lung, but not the heart. And then we looked at prostate cancer, which really loves the bone. And something interesting about prostate cancer is that animal models really lack that response. So you'll see the cells metastasize anywhere else but the bone. But then in humans, they always go to the bone. So we thought that this would be a great place to plug in our model and see if we could recapitulate that clinical response. And so our prostate models all show this really high tropism for bone. And then our breast cancer model, the MDA MB231, shows both bone and liver. Something interesting is that we could also really recapitulate skeletal remodeling. So when cells metastasize, especially to the bone, they basically take over this really favorable niche. And most cancers are osteolytic, so they'll eat away at the bone and take, all the good factors from it. But prostate cancers can also be osteoporotic. That was pretty exciting. And this is in full 3D. We've sectioned multiple tissues and kind of found that it happens often. And then, so you'll see with the osteocalcin, again, we have cytokines that kind of match this osteoblastic versus osteolytic and ALP. And then for osteolytic, they're eating away at that bone. So you'll see in the histology that the bone's being removed, and you also have osteopotin secretion high in those lines. And then some prostate cancers have like this mixed phenotype. So they're osteoblastic and osteolytic. And we were quite surprised that when we looked at this histology, it also has this mixed phenotype where you've got the breaking down and the rebuilding otherwise. Then we added drugs that were known to, kind of prevent skeletal metastasis, prevent very loosely, because nothing really works to prevent skeletal metastasis at the moment. So we use cisplatin first. It's just a DNA damaging agent with a known efficacy. And we can see that it does help reduce the tumor in the tissues. And then we use denosumab, which is used to treat metastatic prostate bone loss and acts on the Rank-Rankl pathway. And here it worked better in the more osteolytic groups, which is expected, and it was great to see. We also use ZA, so bisphosphonate, which is very commonly used for, all bone metastases, and also, again, works to inhibit osteoclast activity. And we can see it, does help with tumor growth in the bone. And then we tried dasatinib, which we were quite excited about. It's a tyrosine kinase inhibitor. Then we didn't see a response. Then clinical trial data came out a bit later showing that also clinically there really wasn't a lot of efficacy. So it was, cool to see that this was mimicked. Then we wanted to take it a step further and kind of figure out how to really benchmark our models a bit better. And so we decided to do organ on a chip and animal models, side by side. And so here with the prostate cancer, looking into what's driving bone metastasis in both mouse and human models. And so at Columbia in the Koryabate Shen lab, they have genetically engineered mouse models that are very metastatic specifically to the bone, especially their NPK model that has that NKX, P10, and KRAS modified. And so you'll see they do metastasize and they do go to the bone, and they very much go to the bone. So it's a highly penetrant bone metastatic mouse model. And so we took cells from these genetically engineered mouse models and put them in our platform to see if we could just replicate what's seen in the mouse. And we did. So they go to the bone, and then you had those varying degrees of metastasis in the different models. So the NP mouse model really doesn't have metastasis. And you'll see in panel C that was also in our system. The NPM has moderate metastasis, also replicated. And then that NPK model was very metastatic to the bone. And so that was able to be replicated, from the animal model within our organ on a chip. And so now we felt kind of comfortable that the model was predictive. And so we started, diving even further and doing organ on a chip and animal models kind of side by side. And so using a CRISPR activation screen, SIDED2 came up as a hit as a novel driver of prostate bone cancer metastasis. And so looking into, overexpressing it and seeing if that can drive metastasis or not, we put these cells both in the organ on a chip assay and in these mouse models and found that, SIDED2 really is a driver of bone metastasis specifically. And then again, so ATAD2 came up, not necessarily being so bone tropic, but just as a regulating metastasis in general. And so again, we did the organ on a chip assay and the mouse model, and we're able to show that it does drive metastatic progression. And then going back to the heart, so we also want to be able to do personalized drug screening. So take your iPS cells and make them into a model. So here we looked into the drug cisapride. So this was a drug that really got popular because it helped with heartburn and GERD. So it kept getting prescribed more and more. And then unfortunately, there were very toxic, lethal cardiac side effects. And so then looking back at it, it passed all of the screenings and made it to market safely. Back when this went to market, there weren't as strict of the preclinical assays that we have now. So it probably wouldn't have made it through, but it turned out that it blocked the hERG channel and had this QT prolongation. So most people taking it, you would be fine. But if you had an underlying disease, you would be more susceptible, or, a genetic mutation that helps you or changes how you would metabolize this drug to have a higher, so that it accumulates to a higher dosage. So people with Timothy syndrome, which is also prolongs the QT, when they take that drug, that happens to prolong the QT, it leads to these electrical malformations that cause arrhythmia, or anybody with hypertrophy would be much more at risk for these arrhythmias. So we can kind of detect, where, what populations drugs should not be given to, and whether they're safe or not. So overall, in summary, we found that maturing the tissues was really important to help us get those tissue-specific drug screening responses, and then connecting them in a way that keeps that maturity helps us enable, as faithful as we can, these systemic models towards clinical translation. And we see this as like a missing puzzle piece to add to the whole drug discovery, drug safety screening landscape. And so on one hand, it can help you with drug development, mechanism of action studies, identifying, whether it's working or not, and is it safe, and then also including more patient diversity into drug screening, as well as personalized drug screening. Does this drug work for me? In the future, we're working on making them more reliable and accessible. So, nobody wants to deal with tubing and all these open systems. So having it on a well plate, and then to be handled by liquid handlers for more automation. So just trying to keep it open, source, modular, but easy to use. I'd like to acknowledge everybody in the GVN Gordana Vanyuk-Novakovic lab and all our funding. Are there any questions?
Can you hear me?
Yes.
Ah, okay. Seems I disappeared for a moment there. Thank you so much. It was a wonderful talk. Thank you, Casey. We do have a couple questions. One of them is barrier functions for individual organs are different. How do you establish that in your multi-organoid system?
Could you say that again?
Given that barrier functions for individual organs differ, how do you establish that in your multi-organ system, or how do you account for that?
So right now we're not necessarily accounting for it at all, but absolutely correct, especially once you get into the lymphatic landscape. So right now we're just adding the cells and they're kind of adapting. You saw that the liver was a little more permeable than all the other ones. And so putting cells in the right environment and letting them do what they do. But we have been putting more work into getting more of this micro to macro vasculature, where you would have a lot more of that modeling incorporated. And, we'll see. So far, we're just putting the cells where they in the right tissues and they're adapting on their own.
Wonderful. Thank you again, Casey. That concludes today's presentations. On behalf of the scientists, I'd like to thank the entire panel for sharing your work. Thomas Durkin, Jasmine Trigg, Joanna Neves, Grace Emin, Natalia Vergara, Derek Sokolowski, and Casey Ronaldson-Bouchard. And our symposium sponsors, PHCBI, Sartorius, Donovix, Sinobiological, BioXcel, Biotechne, Inventia Life Sciences, Stem Cell Technologies, and Lycor Bio. If anyone has any further questions, please consider reaching out to the speakers directly. Their contact information is shown on the screen. As a reminder, the symposium will be archived on the scientist website, and you will receive an email notifying you when the recording is available. Make sure to follow the scientist on social media for updates on future events. I'd like to thank everyone who took the time to join us today, and especially those of you who shared your questions and comments. Thanks, everyone. Have a good day.